Good morning, ladies and gentlemen. With great joy and reverence, we welcome you all to this gathering. Today, as we unite under the banner of knowledge, collaboration, and celebration, let us begin a conference with solemnity and grace. To invoke divine blessings, let us begin with a prayer song, followed by a college song and Thamaltai Valtu. May this set the tone of devotion and dedication for the day ahead. So, hold on to get that shine on me, up your soul, 'cause you got it all in the side of those lungs. Get up and stand up. I'm on my soul. Why don't you get shine up the mirror soon? 'Cause you got it all in the side of those lungs. ನೀರಾರು ಕಡಲು ನಿಲವ ಕೆಡಿಲೊಳಗು சீராரும் அதன் மேல் திகழ் பரத கண்டவது
So, every event begins with a warm word of welcome. To extend heartfelt greetings and set the stage for this conference, I now invite Dr. R. Jasmine, convener and head of Biotechnology, Bishop Paper College, to deliver the welcome address. Please, ma'am.
This is the Lord's doing. It is marvelous in our eyes. With a grateful heart, I welcome you all to this 2 days funded conference. A very pleasant good morning to one and all gathered here. With immense joy, gratitude, and honor, I extend a warm and hearty welcome to each one of you on behalf of the Department of Biotechnology, Bishop Heber College, to this DST and DBT funded national conference on future preparedness and emergency response through biotechnological innovations for a prosperous India. Today marks not just an academic gathering, but a confluence of ideas, innovations, and aspirations that will surely illuminate new pathways in scientific research. At the outset, I deem it my pleasure and honor to welcome our beloved principal and patron, Dr. Princey Merlin Ma'am, under whose able leadership and visionary guidance this institution has set benchmarks in higher education and research. A leader is not only one who knows the way, shows the way, but also goes the way. And Dr. Princey Ma'am embodies this in every sense. We are deeply grateful for your inspiring presence today here, ma'am, in spite of your compelling and busy schedule. On behalf of the entire department, I extend a warm welcome to you, ma'am, and thank you for your presence today. With equal warmth, I extend my respects and welcome to our dynamic vice principals, Dr. GGR Satyasilan sir, Vice Principal Aided, welcome you sir, and Dr. Ravidar, Vice Principal Self-Financing, welcome you sir, and our ever supporting Bursa, Dr. C. Dhanapal, welcome you sir, who continually strengthen the academic, administrative, and spiritual pillars of our institution. Welcome you, sirs. It is indeed a matter of great honor to welcome our distinguished delegates who grace us with their presence and expertise. We are delighted and honored to have with us Dr. Kumaravel Somasundaram, Professor, Department of Cell and Molecular Biology, Indian Institute of Science, Bengaluru, a distinguished researcher whose contributions in molecular biology and cancer biology continue to inspire generations of scientists. Sir, we are much privileged and honored by your presence. Welcome you, sir. We are equally honored to welcome Dr. Yadvinder Singh, Assistant Professor, Department of Botany, Central University of Punjab, Bathinda, whose insightful research and academic contributions bring great value to this gathering. Sir, your presence immensely and your immense scholarly strength adds light to this conference. Welcome you, sir. I also take this opportunity to extend a heartfelt welcome to the Shift 2 coordinator, Dr. Cynthia, and also the judges of oral and poster presentations whose critical evaluation and constructive feedback will encourage and shape the budding researchers presenting their work here. I do also acknowledge the presence of the heads and the faculty members of other department. I also offer my sincere welcome to Dr. Priscilla Suresh, the head department of zoology, and members from the other department. With great joy, I welcome the faculty members and students of more than 40 institutions who have joined online and who are going to present their findings in the form of oral and poster presentation. Welcome you, dear participants. To our presenters who will share their innovative ideas and research findings, I say research is creating new knowledge. In the words of Neil Armstrong, may your work ignite curiosity, foster discussions, and inspire future collaborations. Welcome you all. I extend a warm welcome to the teaching and non teaching fraternity of our own department, my colleagues who have been the backbone without whose support this would not have seen the light. I also welcome my dear students who are the heartbeat of every academic endeavor we undertake. Welcome you, dear students. A special word of welcome goes and appreciation goes to Mrs. Melba Priyadarshini and Dr. Dinesh Raja, the organizing secretaries who were responsible for tapping government funds from both DBT and DST. So, welcome you and thank you for your efforts. I also welcome and thank the editorial team who had worked tirelessly with sleepless nights to bring about the proceedings. Thank you and welcome you, dear team, whose seamless effort ensures the smooth execution of today's hybrid mode conference. Once again, I extend my warmest welcome to all gathered here online and offline. And I pray that this conference will become a beacon of knowledge, collaboration, and innovation. Education is the most powerful weapon which you can use to change the world, said Nelson Mandela. May this gathering of brilliant minds be a step towards changing the world through science and innovation. So, with a grateful heart and a joyful heart, I welcome one and all gathered here. Thank you.
Light symbolizes knowledge, wisdom, and prosperity. To illuminate this gathering with auspiciousness, I request all the dignitaries to come forward for the ceremonial lighting of the Kuthavilaka. Let the glow of the lamp guide us towards enlightenment and success.
It is our privilege to honor the esteemed guest whose presence graced this conference.
To begin with, we would like to honor our respected principal, Dr. Princey Merlin. May I invite Dr. C. Sindhia, Assistant Professor, Shift 2 Coordinator, Department of Biotechnology, to present the shawl to our beloved principal. Please, ma'am. Now I request Dr. R. Jasmine, Convener and Head Department of Biotechnology, to present the gift to our principal as a token of love. Please, ma'am.
Next, it is our great pleasure to honor our chief guest, Dr. Kumaravel Somasundaram, Professor, Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. I request Dr. JJR Satyaseelan, Vice Principal, Aided, Bishop Paper College, to present the shawl and gift to our sir. Please, sir. I now invite Dr. C. Ravidas, Vice Principal SF, Bishop Paper College, to present the shawl and gift to Dr. Yadvinder Singh, Assistant Professor, Department of Botany, Central University of Punjab, Bathinda. Please, sir.
We also take this opportunity to honor our respected Vice Principal and Bursar. I request Dr. J. Dinesh Raja, Assistant Professor, Department of Biotechnology, to present the shawl to Dr. JGR Satyaseelan, Vice Principal, Aided, Bishop Weber College. Please, sir. Now I invite Dr. R. Sharmila, Assistant Professor, Department of Biotechnology, to present the gift. Please, ma'am.
I now request Dr. G. Ganapati, Assistant Professor, Department of Biotechnology, to kindly present the shawl to Dr. Ravidas C., Vice Principal, Bishop Paper College. Please, sir. I also invite Mrs. Pelba Priyadarshini, Assistant Professor, Department of Biotechnology, to present the gift. Please, ma'am.
I now request Dr. P. Samuel, Assistant Professor, Department of Biotechnology, to present the shawl to Dr. C. Dhanabal, Bursar, Bishop Weber College. I now invite Dr. A. Anita Margaret to present the gift. Please, ma'am.
A guiding force, a visionary leader, and a constant source of inspiration. Our principal, Dr. Prince now takes the stage to deliver the presidential address. Ma'am, we are eager to hear your words of wisdom. Please, ma'am.
Eminent scholar from IIC Bangalore, Professor Somasundaram sir, Dr. Edwin Singh, respected Vice Principals and Bursar, beloved Head of the Department, Dr. Jasmine, Shift 2 Coordinator, Dr. Cynthia, the Organizing Secretaries, Dr. Melba and Dr. Dinesh, beloved Heads of Departments of Zoology, Food Science, Bioinformatics, faculty members, student friends, and delegates from various institutions. Good morning. My greetings to all of you. Let me begin by recalling the word of God from Proverbs 24:5. The wise prevail through great power, and those who have knowledge muster their strength. Today's gathering is indeed a reflection of this truth. As we come together to draw upon the strength of wisdom, knowledge, and innovation to prepare for the challenges of our times. It is my profound joy and privilege to preside over the inaugural ceremony of the two-day National Conference on Future Pandemic Preparedness and Emergency Response through Biotechnological Innovations for a Prosperous India. This theme is not only timely, but also visionary, as it speaks to the need of our nation and the global community. My special word of appreciation to the head of the department and the organizing secretaries for taking efforts to organize this need-based national conference. The pandemic we all endured reminded us of our vulnerabilities, but also awakened us to the immense potential of science, technology, and collaboration. Biotechnology, in particular, stands as a beacon of hope, whether through vaccine development, diagnostics, or innovative therapeutic solutions. The focus of this conference encourages to look beyond survival toward preparedness, resilience, and prosperity. As Louis Pasteur once said, chance favors the prepared mind. This conference is a stepping stone in preparing our academic minds and research culture to foresee challenges, respond effectively, and to innovate for a healthier and safer tomorrow. We are deeply honored to have with us today an eminent scholar, Dr. Kumaravel Somasundaram sir, Professor, Department of Cell and Molecular Biology, Indian Institute of Science, Bangalore, as our chief guest. Sir, your pioneering contributions to the field of biology and your presence here will undoubtedly inspire our faculty, scholars, and all the participants. Thank you, sir, for your continued support to our institution. We look forward to your inaugural address with great anticipation. I would also like to appreciate the Department of Biotechnology of Bishop Heber College under the leadership of Dr. Jasmine Jemima and Dr. Cynthia, Mrs. Melba Priyadarshini and Dr. Dinesh Raja took lot of untiring efforts and able to get the fund from the government of India. My sincere appreciation and congratulations to the head of the department, Dr. Mrs. Melba and Dr. Dinesh, and all the faculty members and students of the Department of Biotechnology, Bishop Heber College. Your untiring efforts, meticulous planning, and passion for academic excellence have resulted in successful organization of this national conference, jointly supported by Department of Biotechnology, Government of India, and Department of Science and Technology, Government of India. My wholehearted appreciation, especially to Dr. Jasmine. I warmly welcome all the participants, research scholars, and faculty members across the country and beyond. I am certain that these 2 days of discussions and deliberations will spark new ideas and insights, forge collaborations, and build capacity for future pandemic preparedness. My hearty wishes and congratulations to all the participants from 40 different institutions. With that, I wish this conference all success and pray that God's wisdom and guidance will continue to lead us in all our academic endeavors. Once again, I wish everyone all the very best. Thank you.
Thank you, ma'am. Next, it is time for felicitations. I now request Dr. JGR Satyaseelam, Vice Principal Aided, Bishop Paper College, to felicitate the gathering. Please, sir.
Esteemed chief guest of today's inaugural ceremony of the 2 days national conference organized by the Department of Biotechnology, Dr. Kumaravel Somasundaram sir, who is a professor, Department of Cell and Micromolecular Biology, Indian Institute of Science, IISc Bangalore, respected guest of honor, Dr. Yadvinder Singh, who is from Central University of Punjab, Patinda, respected Principal Madam, Vice Principal, Self-Financing Stream, beloved Buzzer, Head of the Department Biotechnology, the organizers of the 2 days national conference, Dr. Melba Priyadarshini and Dr. Dinesh Raja, Shift Coordinator, Dr. C. Cynthia, other learned colleagues from biotechnology, other heads from other departments. I'm able to recognize Head of the Department of Zoology, the Principal of Suresh Madam, Dr. Rosalind from Food and Nutrition, Dr. Akila from Bioinformatics as heads. They are here. And my dear friends, it is my joy and honor and privilege to be the part of today's inaugural ceremony of this 2 day national conference. As I was coming towards this hall, I already had a few talks with you. We, the computer science people, are dealing with machines, whereas you people are dealing with lives, which is important. We are dealing with artificial intelligence, but you are dealing with real intelligence. Who are intelligent people here? Fine. So the point here is that we, the computer science people, are inspired by the biologically, created things. How come this artificial intelligence emerged? That is because of natural intelligence. How these artificial neural networks emerged? That is because of the biological neural network. That means brain. The study of brain involves and evolves artificial intelligence. I was going through the invitation. Your HOD name is Jasmine. Madam, nice to have. Yeah, madam is there. Yeah. Jasmine means real. But I'm not sure whether this flower is real. It's plastic or real. I don't know. Plastic. I'm sorry, our college is campus free. I should not expose this matter. But dear friends, don't go after artificial things. Go after real things. Because you are people, you are dealing with the real things. You have to spread the fragrance. But I'm looking at the lamp. Lamp is full of real flowers. Dear friends, what is the difference between the artificial flower and the real flower? Both look like the same. They look similar. Problem is that the artificial one, it can also move because of the air, but it cannot spread the smell, correct? Whereas if you go to the real flower, that will spread the fragrance. Jasmine, madam, we consider you as a real one because you have to spread the fragrance of biotechnology in this conference. You are mean. You are real. I am real. I am artificial. I am from artificial background. We are dealing with machines. Machine can do whatever we do, whatever we say. To that. All right. That is number one. Number two, I was thinking about the purpose of this conference. The purpose of this conference is very lengthy. I have to read it carefully. Future pandemic preparedness and emergency response through biotechnological innovations are a prosperous India. A very lengthy one. Very interesting. You are dealing with the diseases of the human being. You are dealing with the human being problems. You are addressing to the issues of the human being. That is fine. That is wonderful. What I want to say as a concluding remark, education is importance. It talks about the importance of life. But you people are doing the importance of life as an education. Education imports the importance of life, whereas you people are inculcating the importance of life as an education. நாங்கள் வாழ்க்கைக்காக பாடத்தை படிக்கின்றோம். நீங்களோ வாழ்க்கையே பாடமாக படித்துக் கொண்டிருக்கிறீர்கள். வாழ்க்கைக்காகவே வாழ்ந்து காட்டுவதற்காகவே உயிர்ப்புடன் துடிப்புடன் நீங்கள் உயிரோடு நீங்கள் பாடத்தை படித்துக் கொண்டிருக்கிறீர்கள். Biotechnology. But I want to tell you, bio is yours, technology is ours. Bio is for you, technology is from our side. Let us jointly run together and win together. The future India is not only in your hands, it is in our hands. Let us run together and win together. God bless you.
Thank you, sir. I now request Dr. C. Dhanabal, Bursar, Bishop Heber College, to felicitate the gathering. Please, sir.
Good morning, everyone. After a powerful talk, I don't know whether I will be able to match up to his oratorial skills. Okay. Without wasting much time on the formalities, dear dignitaries on the stage, faculty from the Department of Biotechnology, HODs from other departments, my dear students from Bishop Heber College and students from other colleges, teachers from other colleges, ladies and gentlemen, once again, good morning to all of you. Okay. Dr. Satyasilan sir is from Computer Science Department, so he talked about the computer science. And I am the only person, odd man out of this group because I don't belong to science stream, I belong to pure arts stream. I come from English literature. Yes, you all know that science makes our life a healthy one and arts makes our life beautiful. Okay? And both the science and the arts are the two eyes of a human being, as you all know. Okay? And as far as a common man, a man out of this field, I understand that as our vice principal said, that you are dealing with the bio. Bio means we all know that it is life. You are dealing with life. And I also listened to our principal madam's felicitation and also the welcome address given by the HOD of this department. And I often listen to these two words as far as this biotechnology is concerned, that you are giving a lot of importance to innovation and you are also giving a lot of importance to research. Okay. And that's why you have organized this seminar, and this seminar is a place for a confluence of ideas, new innovation and new ideas. And all these things are going to happen during these 2 days. But dear students, I would like to ask you one question. Why you are doing research? Why you are after innovation? Okay. And the result or the answer can be given during the time of Corona. I think the biotechnologists played a big role during the time of Corona by bringing out many vaccines. and by saving lives. Okay, so you are doing a wonderful job. That during the time of Corona, or even in the ordinary times, you come out with a lot of innovations. You come out with a lot of remedies. You come out with a lot of medicines in order to make this world, as the seminar topic goes, you want to make this world a prosperous one. That is the aim of the biotechnologies. That is the aim. Okay. So let me put it in the arts way, because I come from the arts background. Okay. So what is the mission of the biotechnologies? What is the ethos of the biotechnologies? What is the ethics of the biotechnologies? Biotechnologists want to make a better world. Biotechnologists want to give a better life. In other words, biotechnologists want to give a disease-free world. And put it in a very short manner. Okay, biotechnologists want to give a peaceful world where there should not be any grief, there should not be any sadness, there should not be any sorrow, there should not be any tears. And this world should be a beautiful world. That is the basic responsibility, basic ethos, or basic ethics of the biotechnologies. Okay. And dear friends from the biotechnology department of Bishop Weber College, as well as from other colleges, I would like to tell that and you are going to make this world a beautiful one. You are going to make this world a prosperous one. Let this first step begin with your department. Let this first step begin with your family. Let there be peace in your department. Let there be peace in your family. Let there be peace in your prosperity in your department. Let there be prosperity in your family. Let there be peace, or let there be what you called a kind of a disease free. Disease in the sense both from your body as well as both from your mind. Your body should be free from any disease. And in the same way, your mind should be free from any disease, because we talk about mind, you talk about the body. So both should be free from any disease. There should not be any narrow mindedness. There should not be any childish activities as far as the scientist is concerned. Okay, so this peace, this prosperity, this remedy should begin with the first step should be from your department, from your family, from your college. Then we will step into making this world a beautiful one, a prosperous one. Okay, so I think this conference will be the first step. This conference should be the first step in making your department a prosperous one, in making your department a disease free one, in making your department a peaceful one. And let this conference pave way for a beautiful world, and a beautiful college, a beautiful family, and a beautiful India. All these things are there, but the first step should begin from you, the individual, from the department. Okay, then only we can make others a prosperous one, other a college is a peaceful one, or the world a peaceful one. I hope this conference will pave way for a beautiful department, a peaceful department, a beautiful college, a peaceful college, a beautiful India, and a beautiful, a prosperous India. Finally, a beautiful world and a prosperous world. I once again congratulate the department head and faculty members, especially the organizing committee members, Dr. Dinesh Raja and Melba Priyadarshini, for organizing such a wonderful program. And I hope this program will be a big change maker in our lives. Thank you for the opportunity once again. I wish you all the best in future.
Thank you, sir. Every scholarly gathering finds its essence in documentation. We are privileged to release proceedings of this conference. I request our principal and patron to officially release the proceedings, while copies will be received by our distinguished dignitaries. No great publication is complete without the untiring efforts of its editorial team. I now invite Dr. Prince Merlin, our principal, to honor the dedicated editorial members. With joy, I invite the editorial team. Dr. Beatrice Valdares, Assistant Professor, SG, Department of Biotechnology. Ms. F. Leelavadi, Assistant Professor. Dr. Bliss Ramya Joan, Assistant Professor.
Dr. Christine Freeman, Assistant Professor, Ms. Amrita Anandi, Assistant Professor, Dr. G. Ganapati, Assistant Professor, Dr. J. Antonio Oxymoron, Assistant Professor, Department of Biotechnology. We are deeply grateful to the eminent judges who have evaluated and encouraged the academic pursuits of the participants. I once again invite our principal, Dr. Princey Merlin, to honor the judges for their invaluable service. I request Dr. Priscilla Suresh, Head, Department of Zoology, Dr. J. Nesarajan, Assistant Professor, Department of Zoology, Dr. Paul Ajit Kumar, Assistant Professor, Department of Botany, Dr. Emmanuel Sahayaraj, Assistant Professor, Department of Botany, Dr. K. Akila, Head, Department of Bioinformatics, Mr. C. Rosalind, Head, Department of Nutrition and Dietetics.
To offer a concise yet comprehensive overview of this conference, we now call upon Dr. Dinesh Raja, Assistant Professor, Department of Biotechnology. Please, sir.
Respected principal, respected vice principals, the bursar of the college, dignitaries on the dais, my head of the department and shifted to coordinator madam and Dr. Melba Piridarshini, the organizing secretary of the event. I thank Lord Almighty for giving me this greatest opportunity to magnify my college name as alumnus of this prestigious institution of higher education. I'm going to present the conference overview of the participant and the discussion focusing on the statistics. The National Conference on Future Pandemic Preparedness and Emergency Response through Biotechnological Innovations for a Prosperous India. It is a prestigious conference because of the dynamic support of two funding agencies, Department of Biotechnology, Government of India, and Department of Science and Technology, Government of India. So the discussion on the focuses on vaccines, diagnostics, and therapeutics for the future health crisis, nanobiotechnology, synthetic biology, genomic, and AI application in health. So the statistics of the participation based on the abstracts and committee details, women participants 55 to 60%, and the outside participant from Tamil Nadu 35 to 40% of contributions from Punjab, Haryana, Jammu and Kashmir, Karnataka, Telangana, and the highlights of the national research of the events. And women from rural background in research focusing on the importance of encouraging women, especially from rural backgrounds, to join research and doing their PhD in national institutes of importance. Schemes like DBT Biocare, DST Women Scientist Scheme, and SERB support programs provide pathways for women in science. The closing note, the conference of FPPRBI'25 is not just an academic gathering, it is a launchpad for young researchers and future leaders in the life sciences. By combining scientific rigor, inclusive, and national leadership of the conference sets the tone for building a healthier, safe, and more prosperous India. Thank you for the opportunity. Thank you very much.
Thank you, sir. Every milestone even becomes more meaningful in the presence of a distinguished chief guest. With great pride, I invite Ms. Amrita Anandi, Assistant Professor, Department of Biotechnology, to formally introduce our honorable chief guest to the gathering. Please, ma'am.
Good morning, respected dignitaries on the stage, colleagues, and dear students. It is my privilege to introduce our distinguished chief guest for today's inaugural session, Dr. Kumaravel Somasundaram, Professor at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bengaluru. Dr. Somasundaram obtained his degree in veterinary medicine from Madras Veterinary College, followed by a master's in biotechnology and a PhD in bacterial genetics from Madurai Kamaraj University. He then pursued his postdoctoral training in cancer genetics in Northwestern University and the University of Pennsylvania in the United States before joining IISc in 1999, where he continues to serve as a senior faculty member. A leading cancer biologist, Dr. Somasundaram's pioneering research focuses on the genetics and therapeutics of glioblastoma, one of the most aggressive forms of brain cancer. His laboratory has made significant contributions in understanding tumor suppressor genes, oncoviruses, glioma biology, microRNAs, and novel therapeutic targets. His outstanding contributions to science have earned him many honors, including the International Senior Research Fellowship from the Wellcome Trust in the year 2004 and the National Bioscience Award for Career Development from the Department of Biotechnology, Government of India in the year 2006, which is one of the highest Indian science awards. He is also an elected fellow of all three premier Indian science academies, the National Academy of Sciences India, the Indian Academy of Sciences, and the Indian National Science Academy. Beyond his laboratory, Professor Somasundaram has been instrumental in building institutional capacity in India. He is credited with establishing the first laboratory for molecular oncology at IISc and contributing to the establishment of genomics facility at the institute. He has led multiple cancer biology programs, including the IISc Initiative on Cancer Biology and Therapeutics, and serves as principal investigator at the IISc Genomics Program in collaboration with CSIR's New Millennium Indian Technology Leadership Initiative. He is also the co-convener of the DBT IISc Partnership Program for Advanced Research in Biological Sciences and Bioengineering. He is a member of the DBT Neurobiology Task Force, a member of the editorial board of Cancer Biology and Therapy, Taylor and Francis, and a former member of the IISc Senate. Today, we are truly honored to have him with us to deliver the inaugural address. Please join me in welcoming Professor Kumaravel Somasundaram.
Thank you, Dr. Anandi. All the audience, please believe me, I did not give such a long introduction information to her. I don't know from where you got this. I only told her, to Dr. Dinesh, I sent the information about where I have studied and what I'm doing right now. So in any case, thank you, Dr. Anandi, for such a long introduction. So good morning to everybody. So first, let me thank Dr. Melba and also Dr. Dinesh Raja, who are the organizing secretaries, for inviting me to this conference. And also, let me thank all the dignitaries on the dais, including your principal, Madam, Dr. Princey Merlin, Vice Principal, Dr. Satyaseelan, Ravi Das, Dr. Ravi Das, and Dr. Danbal, and Dr. Jasmine, convener, and Dr. Cynthia, co-convener, and thank all of them for inviting me to this conference. So let me take a few minutes before I actually go into the topic of this particular meeting. I like this quote from Louis Pasteur, mentioned by your principal, Madam. Luck favors a prepared mind. Frankly, I don't believe in luck. Neither I believe in God. Okay, that is individual's preference. Whether whatever the religion you belongs to, whether you believe in God or not, that is individual's liking, and I don't bother about that. However, I still like this sentence: "Luck favors a prepared mind." We all have to prepare ourselves. We have to work hard. You want to get into Indian Institute of Science for PhD, for example. If I take that as an example and give you an advice, you got to first qualify yourself. You have to do BSc and you have to do MSc, and you have to get very good marks. And also, you have to write one of these fellowship examinations. Like, for example, UGC CSAR entrance examination for JRF, or DBT JRF, ICMR JRF, or GATE examination. One of these examinations you have to write, and you have to pass. Not only pass, you have to get a very high rank, because the number of people who pass these exams go several thousands. Typically, in a year, in our department, we can only interview about 400 to 500 students for PhD program. But there are several thousands of people pass all over India. One of the major problems in our country is the population, 140 plus crores. We have surpassed China a few years ago, right? So while that is a problem, it's also a good thing that we have a huge number of young population. So we have a large number of people passing these exams, but we could only handle few hundred students for interviewing them to select for PhD. Okay, so many students who pass above the cutoff mark, and they called for the interview. About 500 students come. Out of that, we have every department about 12 to 15 students only we can select for PhD program. So if there are 500 students come, we get only 10 to 15 students get selected. Okay, all 500 students are very good students because they all have passed these exams, and not only they passed the exam, they have come above the cutoff. It's very difficult to find any kind of difference in their qualities and their abilities. Still, we have to find some difference among them to choose the best 10 to 15 out of 500. Okay, so there comes this particular sentence: luck favors a prepared mind. While every student has come well prepared, okay, you can just walk into the interview at particular time, morning 10 o'clock, attend the interview. On the other hand, you can just hang around 2-3 days in the place where interviews are held, talk to the students who are coming out of the interview, what kind of questions are asked, what are the answer for those questions. Possibly as a teacher sitting on the other side, there can be 100 questions I can have in my mind, maybe 200, 300. I can't possibly have a lack of questions, right? So if you just hang around and talk to other students who are coming out of the interview hall, you get an idea what kind of questions are being asked. So this comes under the preparedness. Okay, luck always favors a prepared mind, even though I don't believe in luck. But sometimes luck does seem to happen. For example, you want to get ready and go and take a bus, get a bus to go to some place. You know the bus comes at a particular time. Okay, plus or minus, let's say 5 minutes, the bus comes. Or a train starts around 7 o'clock, 8 o'clock, whatever it is. You always prepare yourself. Go 10 minutes before, 15 minutes before. You just make sure you never lose the bus. So that way, in any aspect of our life, including studies, if you prepare yourself, you will always get it. What is only in that sense, I agree with the statement, luck favors the prepared mind. Even though I never ever believe in luck, if you really work hard and do things properly, study properly, you can achieve anything in your life. That is the message I want to convey. Then I will go to the today's aspect of this conference, the future pandemic preparedness and emergency responses through biotechnological innovations and are a prosperous India. And as Dinesh said, it is nicely funded by Department of Biotechnology and Department of Science and Technology. This kind of a workshop is really, we are in need of it. So if you all remember, not long ago, about 5 years ago, do you remember mask? Do you remember hand washing liquid? We forgot. How many of us are wearing? Anybody is wearing a mask here? I'm not sure anybody is wearing a mask. And the mask has become so popular, it has become that particular days normal. It has become a normal of Corona days that everybody has to have a mask. And added to that, masks have come in different colors. Particularly for ladies, it used to come in different colors with the different type of designs. It has become such a kind of a normal routine. Mask means it's an important aspect of our life. Like we have a pen, we have a shoe, a mask is also part of our life. Hand washing liquid. And added to that, hand washing liquid dispenser. Every lab, entrance to a lab or entrance to a hall, entrance to a lecture hall or a classroom, there will be an automatic dispenser. You don't want to use your hand to touch the bottle. You can use your leg to actually press it and it will be dispensed. So the point is, if you remember, the coronavirus, which is about only 100 nanometers, okay, in size, it has taken the entire world for a ride, isn't it? And I still remember March 25, 2020, the first lockdown started. The announcement was made few days before. March 25 was actually a Tuesday, 2020. The Saturday, Saturday or Friday evening, Prime Minister Modi announced the lockdown. Saturday, my laboratory used to have a lab meeting where students all meet and then they present the work, what they are doing. And we were all discussing what to do. And email has been sent to the students who are doing PhD in my lab and all the other laboratories. They have to vacate hostel very next Monday. Only 2 days were given to them. Saturday morning, the mail has come. Saturday, Sunday, and Monday morning, they all have to vacate. People have come from all over India. Okay. It's not even possible to go home within a day. Most students who cannot afford a flight, they have to take a train. They have to take about 2 days to go to extreme parts of North India or western part of the country, eastern part of the country. They used to take 1 day or 2 days to go there. So it was not that an easy thing to remember. Within 2 days, everybody has to go home. And it was like, I think 6 weeks, right? 4 weeks. First one was a 6 weeks lockdown. The point is, even though it has taken the entire world by surprise and taken for a ride, mankind was able to overcome that. If you remember, immediately after lockdown, all the rules were laid down. We need to wash our hands and we should not touch our eyes, right? And keep washing as many number of times as possible. Don't go out. Don't go out unnecessarily. Don't go to places where a lot of people stay. And if you remember, if you go to the railway station, there will be a sitting area. One seat will be empty. There will be one empty seat between two seats. And even in the aircraft, if you are flying, they used to keep one seat empty. I mean, if you look at that, it looks crazy right now. But that was the need of the hour at that time, if you all remember. So subsequently, we were able to understand the coronavirus. Coronavirus actually was first discovered in few months before, 2019 December. Few months before that, the infection was first noted, and then December, it was actually the virus was isolated, purified, sequenced, and then the paper was published in Nature. And then it few months to recognize the problem. I remember I was attending a meeting in Trivandrum, Indian Association of Cancer Research annual meeting conducted by Rajiv Gandhi Center for Biotechnology. I was attending that meeting. It was in the month of February, and there were several participants from abroad, including from Singapore. They are supposed to come, they have cancelled it. If you remember, the problem started in the Southeast Asian countries. It started in China and then started slowly spreading to Singapore, Malaysia, and those places. So perhaps those scientists who are about to come, they realize the importance or the problems which is likely to happen. They cancel their trip to that meeting, and we were all discussing. We didn't know what is happening. It was in February, just not long ago, just 2-3 weeks before the lockdown. Okay, March 25th was the lockdown. I'm talking about end of February. So we were wondering what is going on, why so many people have cancelled and all those things. I went home, and then few weeks later, sometime around March, first week, second week, the world started talking about the epidemic. I mean, the pandemic thing spreading everywhere. And of course, the lockdown was announced, and then we all went through that. We had this vaccine, wonderful vaccine, Covaxin and Covishield. Covaxin was prepared within India by Bharat Biotech, and Covishield, the vaccine was originally made from Oxford University in UK and made in large quantities by Serum Institute from Pune. We all would have taken any one of these vaccine. I think Covishield was used by most people in India, followed by Covaxin. So the point is, I think the preparedness, the pandemic is not new. Okay, perhaps our generation it is new. Exactly around 100 years ago, the flu pandemic happened in 1920s. Okay, around 100 years before, and that is also a virus. 100 years later, the coronavirus has come. But now there are many task forces have been set up to there are few things one has to keep in mind. There are certain things the government can do. There are certain things all of us has to do to make sure how we prepare ourselves for if at all there is a pandemic comes in the near future. The government job is through various task forces, scientists, they will keep doing some screening. It's possible to see, it's possible to predict what kind of viruses which could actually can actually create a pandemic like situation. People keep doing screening for potential generation and spread of that kind of viruses. This will give us some idea little in advance that likely something may happen. That's number one. This has to be done by government agencies in collaboration with such kind of health organizations all over India, perhaps all over all over the world. WHO and other organizations can do that. From our side, we need to make sure that we always keep the very healthy practice all the time. Perhaps using a hand washing liquid, possibly using the mask when you go to a very crowded places. It's still a good practice to always follow. We always used to wash our hands. It is not anything new. Only thing we have to do it many times during the corona. So following all these principles, make sure that each one of us are individually responsible for perhaps preventing such a pandemic in the future from spreading all over the world. Okay. So with that simple introduction, I stop my talk. Thank you very much. I look forward to listen to my talk after the inauguration is over. Thank you.
Thank you, sir, for your inspiring words. No event can come to fruition without the support and cooperation of many. To extend gratitude on behalf of the organizing team, I now invite Mrs. Melba Priyadarshini, Assistant Professor, Department of Biotechnology, to propose the oath of thanks.
Good morning to everyone present here. It is a great privilege and honor for me to propose the oath of thanks on behalf of the organizing committee of this national conference on future pandemic preparedness and emergency response through biotechnological innovations for a prosperous India. First and foremost, I express our heartfelt gratitude to our chief guest, Dr. Kumaravel Somasundaram sir, Professor, Indian Institute of Science, for gracing today's conference and delivering an inspiring address. Your presence and words of wisdom have added immense value to this event. Thank you, sir. We are also deeply honored by the gracious presence of our distinguished chief guest, Dr. Edwinder Singh, Assistant Professor, Central University of Punjab, who has joined us today as a guest. Thank you, sir. We take this moment to place on record our deepest sense of gratitude to our respected Principal Madam for her constant encouragement, visionary leadership, and wholehearted support in this national conference. Madam, your gracious presence at the inaugural session has inspired all of us and set a positive tone for the entire program. We are sincerely thankful to you for inaugurating the conference and for your valuable words of guidance and motivation. Thank you, Madam. We also extend our heartfelt gratitude to our respected Vice Principal, Dr. Satyasilan Sir, Aided Section, for your unwavering support, guidance, and encouragement in making this national conference a reality. Thank you, sir. We express our special gratitude to our Vice Principal, Dr. Ravidas Sir, Self-Finance Section, for your presence and motivating words have been a constant source of strength for the organizing committee. Your presence at today's inaugural session has added grace and value to the program. Thank you, sir. We take this opportunity to express our sincere appreciation to our respected Bursar sir for allocating financial support and invaluable guidance and in organizing this national conference. Thank you, sir. On behalf of the organizing committee, I extend my heartfelt thanks to our respected head of the department, Dr. Jasmine ma'am, whose constant encouragement, guidance, and support have been the backbone of this conference. Your vision and leadership have made this event possible. Thank you, madam. I also take this opportunity to sincerely thank our coordinator, Dr. Cynthia ma'am, for her meticulous planning, tireless efforts, and seamless coordination. Thank you, madam. I extend my heartfelt gratitude to the esteemed members of the editorial board of this conference. Your expertise, insightful reviews, and commitment to maintaining high academic standards have added immense value to our proceedings. We are equally grateful to the distinguished chairpersons of the scientific session. Your willingness to guide discussion, evaluate presentations will enhance the quality of the event. Thank you. I extend our heartfelt thanks to the distinguished judges of our conference. Your willingness to serve, your expertise, and your unbiased evaluation will bring great value to our technical sessions. Your presence itself inspires confidence and excellence among our participants. Last but certainly not least, we thank and encourage our students. You are the reason we gather, the purpose we serve, and the promise of the brighter tomorrow. Thank you all.
Thank you, ma'am. As we conclude today's formal proceedings, let us preserve these cherished moments forever. I request all dignitaries and faculty members to kindly assemble for the photo session.
Set up a screen here.
Yes, sir. Yes, sir.
Have a short tea break. Students, let's have a short tea break. Kindly refresh yourselves and rejoin by 11:10 a.m. sharp for the keynote session 1. Kindly refresh yourselves and rejoin by 11:10 a.m. sharp for the keynote session 1.
Dear students, kindly assemble. Please be seated as the keynote session 1 is about to start. I repeat, dear students, kindly assemble back in your chairs as we are going to start the keynote session 1. Kind attention to our students at the back. Hurry up, please. Have your teas fast. Please come back to your seats as we are about to begin the keynote session 1. Now we invite Dr. P. Sandam, Assistant Professor, Department of Biotechnology, at keynote session 1. Heber, we now begin oral and person. Please, Dr. Una. I request the class teachers to kindly ensure that the students are seated in their seats. Respected class teachers, I request you to kindly request the students to assemble in their respective seats. Students who are still at the back, I request you to hurry up, have your tea fast, your refreshment fast, and please be seated in your respective seats as we are going to begin the technical session shortly.
Dear class representatives, kindly post in your official group and said that all the students must be present here. I request the students of those who are sitting in backside, kindly come and occupy the front row. Please, sir is going to give an effective talk. It is very useful for us. Kindly come and sit in the front row and kindly post in your class group that the attendance will be taken now.
Check.
We now move on to keynote session 1. I now invite Dr. P. Samuel, Assistant Professor, Department of Biotechnology, to chair the keynote session 1. Please, sir.
Good morning to one and all present here. First and foremost, thanks to the session's resource person, Dr. Kumaravel Somasundaram sir, who is all the way from Bangalore to enlighten us regarding the secrets of dark genome, non-coding RNA component. As you see the title in the screen. Don't get excited, you are alone in the side of dark genome, because SAR is with us to enlighten every one of us regarding the dark side. So don't worry. SAR is about to address several things which I have concised into 5 important points. The first thing is long non-coding RNAs. You oftenly see in the abbreviation as LNC RNA molecule, which was once considered as a junk DNA. Now, with the efforts of several scientists like our professor, it is also found that the junk DNA also has functional roles in the genome, like regulation and cancer-related signaling pathways. And there are many therapies are evolving day by day. Even you could notice Russia has found out a vaccine for cancer, which will come in the years to come. And despite progress in cancer therapies, cure rates remains low, highlighting the need for innovation, particularly in LNC RNA-based approaches. Third thing, high throughput sequencing has revealed LNC RNA as tissue and cancer-specific regulators, making them promising therapeutic targets. The study identified, as per the report or as per the effort of SARS lab, the study identified glioblastoma specific LNC RNA, particularly in glioma stem cell-like cells, which drive tumor progression. A novel non-coding RNA, PITAR, derived from FAM'25B1, all these things will be explained by our professor, was found to inhibit the brake pedal, P53, through a non-genetic mechanism. See, I'm sure these things will definitely feel more complex. As I mentioned earlier, do not worry about these complex things because Torchlight is with us, who is none other than our professor, Kumaravel Somasundaram sir. He will enlighten us regarding the dark secrets of the genome. Sir, the stage is yours.
Thank you. I have. Good morning, everyone. So I'm going to tell you some secrets. That's what Professor Samuel told you, right? Yes, I'm going to tell you some secrets, but I'll make it as simple as possible. You will understand all of them. Okay. So before I actually go to the secrets of the genome or the dark genome, I want to take you out, present some 3-4 slides, introduce some fantastic scientists the world has produced. Okay. So before I go into the genome and the dark genome and the secrets about it and the role of non-coding RNA component, I'm going to make a diverse little bit, take you to this particular slide. You all have studied central dogma of molecular biology. Who has not studied about this? Can you raise your hand? Who do not know what is central dogma of molecular biology? Everyone knows, right? Nobody is raising the hand. That means everyone knows, right? Central dogma of molecular biology was proposed in the year 1958 by Francis Crick. The dogma tells the information flow from DNA to protein is unidirectional. DNA is our genetic material, right? DNA makes DNA through replication, and then DNA also makes RNA through transcription, and RNA undergoes translation to make protein. This is how the information flow happens from DNA to protein, and it is unidirectional. This is what proposed in the year 1958 with the current knowledge at that time what was available. This particular dogma was proposed. Soon, the years passed by, people started asking questions. Does the central dogma still stand? Because there were new discoveries being made in 60s and 70s that central dogma doesn't really seems to stand good. And there were questions, central dogma is empirically inadequate no matter how we see it. Because there were several new findings which doesn't seem to fit into central dogma, which is information flow is unidirectional. I want to show you 2 examples of new findings at the time 70s and 80s, which did not fit into central dogma. Number one, again central dogma is coming. Number one deviation, DNA makes RNA, RNA makes protein, right? The discovery of the enzyme called reverse transcriptase that made possible that RNA can be used as a template to make DNA. So DNA to RNA, it's possible to make DNA from RNA. Reverse transcriptase was discovered. Okay. The great scientists who discovered reverse transcriptase, their pictures are going to come in the next slide. And also one more enzyme was identified, RNA dependent RNA polymerase. Okay. Normally RNA is made from DNA. DNA is used as a template, and RNA polymerase uses DNA as a template to make RNA. That's how the RNA is made from DNA, using DNA as a template. This particular enzyme, RNA dependent RNA polymerase, utilizes RNA as a template to make RNA. These are the two major discoveries the 60s and 70s made, which totally reversed the way we thought how central dogma works. Okay, so with that, I'm going to the next slide. These three gentlemen, unfortunately, none of them are alive today. David Baltimore, Renato Delbecco, and Howard Temin. All these three people, some way or other, contributed to the discovery of reverse transcriptase. Renato Delbecco is a much senior professor. Incidentally, David Baltimore and Howard Temin at some different time points worked as students with Renato Delbecco. Renato Delbecco and Howard Temin passed away quite long ago. And David Baltimore, one of the discoverers of reverse transcriptase, passed away just few days ago, September 6th. Okay. And you won't believe the age at which David Baltimore got the Nobel Prize. Anybody can guess? Typically, Nobel Prize is given to scientists usually in their fag end of their career, 50s, 60s, or 70s. Can any one of the students here, you can guess what age David Baltimore got the Nobel Prize for the discovery of reverse transcriptase? Can anyone guess? Can you get up and guess what age he would have got the Nobel Prize? He was 36 years. David Baltimore was 36 years when he got the Nobel Prize. And without any doubt, the discovery of reverse transcriptase has changed the way biology is done. Okay. So there are any number of experiments we use reverse transcriptase. Even though reverse transcriptase is an enzyme made by retroviruses, which are RNA viruses. These viruses carry RNA as their genome. When they infect the cell, for them to make more RNA genome, reverse transcriptase will convert their RNA to DNA. The DNA goes into the nucleus, gets incorporated into the genome of the host. Then it gets transcribed by the polymerase, that is DNA, RNA polymerase of the host. The RNA viral RNA genome is made in large quantities, and then the virus is assembled and the virus comes out. So the reverse transcriptase, which truly belongs to the retroviruses, is responsible for converting viral RNA to DNA, which then gets integrated into the genome of the host, and subsequently RNA genome is made and virus is produced. So reverse transcriptase was discovered like that. Today in real practice, almost in every aspect of molecular biology, reverse transcriptase is used. For example, if you want to clone a gene, okay, coding gene, only the coding part of the gene, you can isolate total RNA, use reverse transcriptase to make complementary DNA, and complementary DNA can be cloned into expression vectors, and you can isolate your own gene of interest. So every student in my lab or any molecular biology lab, reverse transcriptase is always used. Okay, and if you remember, during Corona pandemic, PCR test is very popular, right? Reverse transcriptase is used there. Total RNAs isolated from the fluids collected from our nose, correct? RNA is purified, correct? And then the RNA is converted to cDNA. Then the PCR was carried out. Reverse transcriptase is used. most of the diagnostic tests where RNA is starting material, reverse transcriptase is used. So reverse transcriptase is so widely used. Okay, this gentleman, David Baltimore, who got the Nobel Prize the year 1976, I forgot. He was hardly 36 years old. He passed away just few days ago, September 6. Perhaps as a respect to this gentleman, I had a chance to listen to him some 10, 15 years ago when he came to Indian Institute of Science. So great man, he passed away. Why don't we show our respect by showing some silence for 30 seconds to Professor David Baltimore. Can we spend some 30 seconds silence? Thank you for 30 seconds. Thank you. Okay, let's go to the title. Secrets of the dark genome, non-coding component. Let me start with the genome first, then what is called dark genome, and then I'll try to explain what are the secrets and specifically expand on non-coding RNA component. Okay, so you all know human genome consists of 23 pairs of chromosomes, right? Our genome is made up of DNA. DNA is present in the form of a chromosome. Each chromosome basically has a long DNA covered by proteins, that is histone proteins, right? 23 pairs of chromosomes are there. Out of the 22 pairs are autosomes, and the 23rd pair is a sex chromosome. So females have a 2X chromosome, males have a X and one Y chromosome, right? Is it understood? It's a human genome, right? So little bit more about it. So in principle, if you remember the central dogma I explained to you, the basic unit of DNA is a gene, correct? A gene has promoter to which RNA polymerase binds, and then it transcribes, RNA is made. The DNA contains the eukaryotic genes, contains exons and introns. Together are transcribed to make a primary RNA. RNA undergoes the process called splicing, during which the introns are removed, exons are stitched together to make a mature mRNA, which undergoes translation, right? Ribosomes carry out the translation to make the protein. This is what typically we all know. Gene is the basic unit of the DNA, right? And typically gene contains a promoter followed by region which is transcribed that makes a primary RNA. Primary RNA contains exons and introns. Introns are removed through splicing. Exons are stitched together to make the mature RNA, which undergoes translation to make the protein. Proteins are the workhorses and they carry out all the work in our cells. Now, these genes are called protein coding genes because they have the capacity to code for a protein. Okay, if a RNA codes for a protein, can anyone tell me what is the most important thing for RNA to code for a protein? Tell me at least one important feature which makes an RNA capable of coding for a protein. Transcription makes RNA, undergoes splicing, exons come together, make the mature RNA. Mature RNA undergoes translation. The RNA, mature RNA should have some features. Remember the title, non-coding RNA, right? That means there are RNA which cannot code for protein. I'm going to talk to you about that. Before I introduce non-coding RNA, you should know what are the features of coding RNA. Then only we can understand why there are RNAs which are not coding. So for you to make it simple, if RNA is capable of coding for a protein, the meaning is that you should have an AUG, start codon, where start first amino acid, methionine can be added, followed by triplets of codons, each one representing different amino acid. And at some point, translation has to stop. You should have a stop codon, where this no tRNA can bind, but it will not carry any amino acid. So a coding RNA should have the coding capacity, which refers to presence of a start codon followed by triplets and end with a stop codon. This is a typical feature of coding RNA. In addition, of course, before the start codon, there are certain sequences to which ribosome binds. In the case of eukaryotic mRNA or coding RNA, these are called Cossack sequences. These are specific sequence to which ribosomes first find and then scan for the presence of start codon AUG. Once the start codon is found, translation starts and it goes on and stops once it sees the stop codon. This is how the translation occurs. Now, remember, consider there is an RNA, there is no Cossack sequence, certainly there is no AUG, so translation cannot happen. Even if there is a Cossack sequence, if AUG is not there, start codon cannot initiate a translation. That's all. Non-coding RNA is just simply like the protein coding RNA, except that it doesn't have the capacity to code for RNA because it doesn't have a AUG start followed by triplet and stop codon. So non-coding RNA is nothing but same as a protein coding RNA, except it doesn't have the capacity to code for a protein because it doesn't have the AUG followed by triplet and stop codon. That's all the difference. Otherwise, absolutely no difference. It is just an RNA as much as a protein coding RNA. I hope you understand. In biochemical sense, this is the meaning of non-coding RNA. Now let's move on. So now we know the whole genome, how big it is, human genome. Thanks to high throughput sequencing, in the year 1990, the whole genome sequencing was started. In the year 2001, about 10 years later, first draft of the human genome was published. Now, after almost 20 years later, 2023, complete draft was published. In the year 2001, quite a proportion of the genome could not be sequenced at the time. The first draft had several gaps. Sequencing could not be completed. 2023, they have completely sequenced the entire genome. Not even a single gap was left behind. So the total genome size is about 3.2 gigabyte base pairs. Otherwise, 3.2 billion base pairs. So this corresponds to one set of genome, not two sets of chromosomes. That is 23 chromosomes, not 23 pairs of chromosome. Okay, this is the size. Now, I'll go to the next part. So whatever I spoke to you just now, protein coding genes, which codes for RNA, which codes for protein, right? So the number of genes which corresponds to protein coding genes belongs to only 2% of the genome. Out of this 3.2 billion base pairs or 3.2 gigabases corresponding to 23 chromosomes, one set of chromosome, only 2%, in fact less than 2%, about 1.5% alone code for RNA, which codes for protein. Okay, so that means what happens to the remaining 98%? In 30s and 40s, I mean 30, 40 years ago, in 16s and 70s, people thought that the remaining 98% has no functions. And people have proposed the terminology called junk DNA. They thought this 98% has no value. It has no function. To the extent people thought you can actually eliminate. Just keeping the 2% alone, one can create a human cell fully functional. Slowly the idea changed. Thanks to the high throughput techniques, when people started sequencing RNA, which is expressed from the genome, they found out that the remaining 98% do seem to have a lot of important informations. Okay, but then yet to be discovered. The so-called junk genome concept changed to so-called dark genome. That is, there is something is there, we don't know what all there. So from junk, the genome 98% has become a dark genome. That there are a lot of elements are there, very useful perhaps, but we have to identify. But today we know that 98% the so-called junk genome once upon a time, later it became a dark genome, seemed to have several elements like transposons, retrotransposons, introns, promoters, telomeres, and pseudogenes. Like this, several elements which are apparently very, very important. Many of them functions are yet to be identified. They all have been mapped to the remaining 98%. More importantly, if you see the next slide, among the 98%, 80% of the 98% also transcribed. That means RNA is not only made from the 2%, which makes protein coding RNA, remaining 98%, 80% of them undergoes transcription. RNA is being made. The only difference is this RNA coming from the 80% of the genome codes for RNA, which does not code for any protein. Okay, these are called non-coding RNA, which can be broadly divided into two types. Those RNA which are less than 200 bases and more than 200 bases, small RNAs and large RNAs. Okay, and then functionally they can also be divided into two categories: housekeeping RNA like rRNA, tRNA, which are involved in translation. On the other side, regulatory RNAs, which can be also divided further into small RNAs, for example, microRNAs. Okay, microRNAs are small RNAs which also regulate the expression of the genes, right? Who got Nobel Prize last year for microRNA discovery? I had a chance to give a talk a couple of months ago when I came here to Bishop Heber College on microRNA discovery. Victor Ambrose, Gary Raukin, two gentlemen from US got Nobel Prize last year for physiology or medicine for the discovery of microRNAs. On the right side is long non-coding RNAs, which are typically more than 200 nucleotides long. Today, I'm going to talk to you about long non-coding RNAs, which are more than 200 nucleotides in length. Now, this slide, I'll skip it. Okay. So as I said, on the left side of the slide, you can see long non-coding RNAs are simply looking like a coding RNA. Okay. You see, for example, they are more than 200 nucleotides. Typically, protein coding RNA also very long. Transcribed by RNA polymerase to 5' end is G-capped and 3' end is polyadenylated. Exons or introns are there. Introns are removed by splicing. Exons stitched together to make a long non-coding RNA. The only difference is they don't have the capacity to code for a protein. I already explained to you why they don't code for a protein. They don't have a start codon and triplet codon subsequently followed by stop codon. That's the reason why they do not have the capacity to code for protein. So long non-coding RNA by all means looks similar to protein coding RNA, except that they don't have the capacity to code for protein. And on the right side, I don't want to explain how the non-coding genes can be present. It can be present between two coding genes or in a sense or antisense orientation to the protein coding gene. They can be located on the genome in variety of ways. Okay, the slides are free. I will leave it here, freely distribute to them to understand more about it. Okay, now let's go to the next slide. So non-coding RNA is not new to you. All of us know about transfer RNA and ribosomal RNA. We all have been studying how the translation occurs, right? The small subunit and large subunit come together and mRNA is loaded onto that, right? And then the start codon, methionine is recognized by methionine tRNA, start codon, and then methionine is added, followed by triplet codon are recognized by the subsequent tRNAs to load the next amino acid one after other. There is a peptide bond formed between the two successive amino acids. That's how the translation occurs. So non-coding RNA is not something new. We know about tRNAs and rRNAs. These are RNAs which do not code for protein. More importantly, I want to introduce one more non-coding RNA, which many of you may not know, but you know the function. We all know male cells, human cells from males have 22 pairs of autosomes and one pair of sex chromosomes. which is one X and Y chromosome, right? Whereas cells from females, we have two X chromosomes, right? So now we know that among the two X chromosomes, one X chromosome is always inactivated. You might have studied about bar body, right? Lyon hypothesis. So what it actually refers to is one of the X chromosome has to be inactivated to have female as a normal female, because one X chromosome expressed genes are sufficient to make a normal human being. That's perhaps the reason why males have only one X chromosome. If in fact, if we have more than one X chromosome, so the dosage of X chromosome mediated genes expression will actually affect our normalness. That is why one of the X chromosome has to be inactivated, and that is done. Inactivation of the one of the two X chromosomes in female cells is done by none other than a non-coding RNA. That RNA is called cis RNA, X inactive specific transcript. Can you believe? Even though this particular RNA is very long, several kilobases long, but it doesn't have any coding capacity because it doesn't have a AUG followed by triplet codon and stop codon. So it cannot code for protein, but it does the most important thing for a female to be a normal female cells. It actually patches, it just goes over one of the randomly selected X chromosome, completely makes the entire X chromosome very compact, and it doesn't allow the transcription to take place. So only one X chromosome is functional. So these two examples, you can understand how the important role played by the non-coding RNA. Yes, coding RNA are very, very important. Coding RNA makes protein. All the proteins in our body, including like RNA polymerase, DNA polymerase, so many enzymes in our body, they are all made by the coding RNA. No doubt about it. But non-coding RNA also plays very important roles. Two best examples I told you. One is rRNA and tRNA, which is involved in making the protein itself. If tRNA and RNA is not there, translation cannot take place. Another best example is a non-coding RNA, which is a long non-coding RNA, which is doing a very, very important function of preventing the expression from one of the X chromosomes. Thereby, the female cells appear normal. Okay, so these are the two examples. The next slide, I will go to the real long non-coding RNA. I gave you two examples, but then how different ways long non-coding RNAs can function. I told you it doesn't have the capacity to code for protein. It is made up of an RNA, but it has no capacity to code for protein. What are the various ways by which long non-coding RNAs function? On the left side, you can see using the complementarity of its RNA sequence, it can bind to DNA, portions of the DNA, if there is a complementarity. It can also bind to other RNA. It can be a protein coding RNA. Using complementarity, it can bind to protein coding RNA. It can also bind to microRNAs, or it can actually carry a microRNA as a host and then allow the microRNA expression to take place. Basically, the functions on the left side corresponds to the ability of the sequence what the link RNA carries. Using the sequence of the link RNA, it can either target a DNA, RNA, or microRNA, or it can act as a host for microRNA. On the other hand, there are certain functions link RNA can do based on the structure. Based on the sequence what they have, they can form a secondary tertiary structure. A scaffold can be made. That scaffold can actually bind to certain proteins, small molecules, okay, or peptides, thereby regulate the function of those molecules, whatever they suppose normally do. So like this, there are multiple ways by which long non-coding RNA can carry out their function when they don't have a capacity to code for protein by themselves. Today, I'm going to talk to you about a particular link RNA which binds to a specific protein coding RNA and how it regulates the metabolism of that protein coding RNA. Thereby, the translation of that protein coding RNA is regulated, and whatsoever the function that particular protein can do normally is also regulated. That is the story I'm going to talk to you about. So I hope you are all able to follow so far. Is it difficult for you? Are you able to follow? Right? Yes? Okay. Now let's go. So what I'm going to talk to you today, in our lab, in my lab, we discovered one link RNA. It is called PITER, P-I-T-A-R, PITER. And we discovered this particular link RNA is highly expressed in brain tumors. My lab works on genetics of brain tumors. One of my students found several link RNAs are very highly expressed. The levels of the transcript is elevated in brain tumors compared to the normal brain. And one such link RNA was Peter. And he went on to do experiments to prove the Peter link RNA can inhibit the function of the protein called p53. How many of you know about p53? Can anyone raise your hands who knows about p53? Anyone? Okay, here is one hand. p53 is biochemically, it's a sequence specific DNA binding transcription factor. It binds to DNA, activates transcription. We talked about RNA is made from DNA by transcription, right? So proteins like p53 along with the RNA polymerase binds to DNA on the promoter, activate transcription. Biochemical sense, p53 forms a dimer of dimer. Two dimers come together, form a tetramer and binds to DNA. You can see on the left side rotating thing, the middle, the orange one is actually the DNA double helix. The tetramer binds to it and activates transcription. Okay, so our finding is Peter link RNA, which has no capacity to code for protein, actually inhibits p53. I'm going to explain to you how we made this discovery. Okay. So let me introduce p53 first and then come to Peter. p53, as I told you in the previous slide, it forms a tetramer, binds DNA on the promoter of a particular gene, activates transcription. There are certain genes which activates, it brings about cell cycle arrest or programmed cell death, otherwise called apoptosis, is carried out. But then if the p53 is always activated like this, it is not good for the cells, correct? Cells will get arrested or cells will die. So it's not good for the cells to have so much protein, p53 all the time in the cell. So that's why p53 levels are kept very low under normal circumstances when it is not required. Only when there is a stress to the cells. Stress can happen due to variety of reasons. For example, when the cells are exposed to DNA damaging agents, then there is a stress. p53 recognizes that and the level increases and that activates certain genes, brings about cell cycle arrest. It doesn't allow the cells to go through cell cycle, arrest them in one particular stage, allowing sufficient time to repair the damage. And then cells can again go back to the cell cycle. Okay, this is what normally how p53 is regulated and how it carries out the function. But what happens in the cancer? In the cancer, p53 gets mutated. Mutated means some amino acid converted to a different amino acid. Okay, so because of that, two things happens. One, the mutation makes automatically p53 level to increase. Okay, in the normal cells, p53 is wild type. The levels are kept very low. Only when there is a stress, p53 level increases and then does this function. Whereas the cancer, when it is mutated, mutation leads to automatically the level increase, number one. Number two, it also forms tetramer, but it cannot activate transcription because mutation occurs in the DNA binding region. You see the picture on the right side? The green color area, the middle, that corresponds to DNA binding region. Most of the mutations occurs in the DNA binding region. Because of that, even though it forms a tetramer, it cannot bind to DNA, it cannot activate transcription. Now let's go back to the normal cells. I told you in the normal cells, p53 levels are kept very low because if it is active, it will kill the cell. It is not good for the cells. Cells want to undergo replication. So it doesn't want to get halted. So p53 levels are kept very low. Only when there is a stress, the level increases, tetramer is formed, binds to DNA, brings about cell cycle arrest to allow sufficient time to repair the damage. If the damage is too much, the cell undergo programmed cell death or apoptosis and cell dies. This is what happens. So when damage happens, when there is a DNA damage, p53 gets activated. How to bring back the normalcy? How to come back to the normal level of p53 so that cells can be there happily? So that is done by another circuitry where in addition to those two genes, one more gene called MDM2 which is also a target of p53, which also gets transcribed. MDM2 protein is made. MDM2 protein is coding for a E3 ligase. Its function is a E3 ligase. It adds biotin, not biotin, it adds ubiquitin to p53 and promotes ubiquitin dependent degradation. Thereby, the p53 level comes down, the cells come to the normal condition. So this is how p53 DNA damage response gets activated and MDM2 brings back to the normalcy. Okay. Our finding is the long non-coding RNA Peter binds to a protein coding RNA called TRIM28. Okay. And TRIM28 RNA codes for the protein called TRIM28 protein. The TRIM28 protein also has the same function as the MDM2 protein. This also adds ubiquitin moiety to p53 and promotes its degradation. So basically, MDM2 and TRIM28 protein together ubiquitinate p53 and promote its degradation. So basically, by binding to TRIM28 RNA, Peter is able to inhibit p53 function. This is what our finding is. The Peter stands for the expansion for Peter is p53 inactivating TRIM28 associated RNA. This finding was made by my postdoc, Dr. Samadhi Jana. Okay. So our lab, as I said in the beginning, middle of the last 5 minutes, work on brain tumors. Okay. Brain tumors, one of the aggressive brain tumors, glioblastoma, which is a grade 4 glioma. Glioma is actually tumor of glial cells in the brain. It's one of the very highly aggressive tumors. So some of the informations are there. I will skip this slide. So Peter, how did we identify Peter? So we had earlier published a paper some 7-8 years ago where we took a large number of brain tumor samples. We also had some normal brain sample. We isolated RNA and tried to do RNA sequencing, identify differentially regulated protein coding RNA and also a long non-coding RNA, and identified several long non-coding RNAs which are differentially regulated in the brain tumors compared to the normal brain. And from that, we identified one link RNA which is called Peter, which is about 4.2 kb in length. It has 4 exons and 3 introns. So as I told you before, it is like a protein coding RNA. Introns are there, it undergoes splicing, exons are stitched together and make a mature Peter RNA. Okay. And to make the story start, some facts, whatever we established, I'm telling you here. Peter is highly expressed in glioblastoma. Okay, upregulation seen only in the brain tumors, not in any other tumor. Interesting. Okay. And if you look at the normal tissue in our body, interestingly, it is expressed very highly only in the testis, not in any other tissue. Okay. And we found this particular RNA is located in the nucleus. Okay. And when you take brain tumor derived cells where a lot of Peter is expressed, if you silence the expression using RNA interference, the cells do not grow. It brings the cells to arrest. Cell cycle arrest happens in G1 phase, and this inhibition happens long time. Peter expression is inhibited for a long time. Cells undergo death. But if you do the same thing in the normal brain cells, normal glial cells, it doesn't affect the growth. Only in the brain tumor derived cells where the Peter levels are higher, if you inhibit the expression, it inhibits the growth of the tumor cells. Okay. Now next slide. So as I said, non-coding RNA do not code for the protein. It does the function variety of ways. It can bind to DNA, RNA, or microRNA, or it can act as a host for microRNA, or it can bind to protein, small molecules, or metabolites and regulate variety of ways. So we have now identified Peter to be highly elevated in brain tumor, and its expression is very important for the growth because if you silence the expression, cells die. So our job now is to find out what is those several pathways by which I told you long non-coding RNA can function. What is the target of this link RNA Peter through which it carry out the function? Okay. So for that we used 2 approaches. One, RNA can bind to RNA, right? So if RNA is bound together, if you bring down one RNA, RNA bound RNA also will come down. So what we have done is we have made 14 antisense oligonucleotides. You see that purple color oligonucleotides which are tagged with the biotin molecules. So we made 14 antisense oligonucleotides. Okay, antisense means which are complementary to the link RNA Peter, biotrinator antisense oligonucleotides, and then using streptavidin beads to bring down the Peter RNA from the total RNA extract. When you bring down like that, all the other RNAs which are bound to Peter also will come down. Thereby, one can identify Peter bound RNA. Is it clear? So, by bringing down the Peter RNA using biotilated antisense RNA, you can find out the RNAs which are bound to Peter. So, Peter bound RNA can be identified. That is bound targets. We identified several of them. We made a prioritization. We found there are about 15 targets. You see that small scatter plot at the bottom. 15 targets which are RNA targets which are binding to the Peter. We took it for our next study. We have to choose one of the 15 to study. Perhaps that is through which the Peter may function. On the other side, we also carried out total RNA sequencing after silencing Peter. Let's say Peter level is very high to begin with in glioma tumor cells. And then when you inhibit the expression, the cells die, right? So by comparing regulated RNAs, regulated protein coding RNAs in the presence and absence of Peter, one can actually find out what kind of functions are regulated in the absence of Peter. By that particular method, we found out P53 protein is probably regulated. The functions of P53 protein is probably regulated by Peter. So we might try to connect these two things. We have 15 targets, 15 RNA targets to which Peter binds on the left side experiment. Right side, the experiment demonstrates P53 molecule may be regulated by Peter. So by connecting these two, given the known function of P53 to inhibit growth, given the known function of Peter to promote growth, because when you silence Peter, it affects the growth, right? That means normal function of Peter, it is promote growth, right? So by comparing these two studies, we found out one of the 15 genes was TRIM28 RNA. TRIM28 RNA coded protein is an inhibitor of P53. Given that P53 inhibits growth, TRIM28 inhibits P53 and the Peter RNA binds to TRIM28 RNA. Peter is a promoter of cell proliferation. Putting together, we made a hypothesis that Peter RNA may bind to TRIM28 RNA and does something to TRIM28 RNA stability, promote the production of more amounts of TRIM28 protein, and thereby it will go and inhibit P53. This is our working model. And we went on to show a lot of data put together that Peter transcribed, Peter is actually present in the nucleus, number one. Number two, we confirmed the interaction between Peter RNA and TRIM28 RNA, we confirmed, number one. Number two, number three, the interaction increases the stability of the TRIM28 RNA. When Peter is there, Peter binds to TRIM28 RNA and increases stability of the RNA. And when there is more TRIM28 RNA, more TRIM28 protein will be made. And what is the function of TRIM28 protein? It also acts as a E3 ligase. It will promote ubiquitination of P53 and promote the degradation of P53, right? So, and we also mapped the interaction domains in the Peter RNA and also in the TRIM28 RNA. Next slide. How do we prove? So now, so far we have found out Peter is essential for the growth and Peter may target P53 and it may do so by interacting with the TRIM28 RNA. We map the interacting domain between P53 RNA and Peter RNA and TRIM28 RNA. The interaction increases the stability of TRIM28 RNA. More TRIM28 protein is made, which will go and inactivate P53 through increasing the proteasomal degradation of P53. So we need to prove by doing additional experiments. Okay, how we do that? The next slide you can see. Let us consider to start with the brain tumor cells where the levels are very high for Peter, which means RNA will bind to TRIM28 RNA, increase the TRIM28 RNA stability, more TRIM28 protein, and that will keep the P53 protein level less. Is it understood now? In brain tumor cells, To begin with, the levels of Peter is very high, which is a non-coding RNA, which binds all the molecules of TRIM28 RNA and increases stability of TRIM28 RNA. So that means more TRIM28 protein, which will keep the p53 degraded all the time. That's good for the cells to proliferate freely. Because you remember, very first slide I showed you, 2-3 slides later before. If p53 levels are very high, it will inhibit the growth. So by this way, in the cancer cells, brain tumor cells, high levels of Peter will increase the TRIM28 RNA, in TRIM28 protein, keep the p53 protein low. Cells can grow very nicely. Now, how do we confirm that? The levels of Peter is very high. That makes the cells to proliferate because it will keep the p53 protein less. So let us say we silence Peter, right? If we inhibit the expression of Peter, automatically the Peter level will come down, TRIM28 RNA will come down, TRIM28 protein will come down, p53 will increase. That will bring about cell cycle arrest. That's what exactly on the top side I'm showing. When you silence Peter, TRIM28 Peter RNA comes down, TRIM28 RNA comes down, TRIM28 protein comes down, p53 protein level increases, and transcription increases, brings about cell cycle arrest. On the other hand, Peter is very high. If you express even more higher amounts of Peter, the TRIM28 RNA will increase, protein will increase, and p53 level will come down. So, silencing Peter will activate p53, overexpressing Peter will inhibit p53. Okay, this we are able to demonstrate. I've shown the data there. Now, is this process silencing Peter activates p53, overexpressing Peter inhibits p53, but these two things are happening with the intermediary, that is TRIM28 RNA. Okay, so now one can actually prove that silencing Peter activates p53. That is because it is not able to target the TRIM28. So if I overexpress TRIM28 under the conditions where Peter is silenced, so p53 activation will not happen. We are able to prove that. Similarly, overexpression of Peter on the bottom side will increase the TRIM28, thereby it will inhibit p53, right? That also does not happen if you simultaneously silence TRIM28. So these set of experiments prove indeed Peter regulates p53 through its action on the TRIM28 mRNA. Okay, we also confirmed when this happens, p53 half-life increases in Peter silent cells because its stability increases. So basically, the half-life is increasing. On the other side, ubiquitination increases when Peter levels are very high. These are some confirmation experiments to confirm indeed what we are showing is correct. Now, more importantly, p53, I told you, when we introduce p53 normally, p53 is present in small quantities that gets activated only when there is a DNA damage. So far, I showed you endogenous p53, which is in small amounts, their function can be regulated by Peter levels. We also show DNA damage activated very high levels of p53 can also be regulated by Peter. So these set of experiments we identified that Peter also inhibits DNA damage induced high levels of p53, number one. Number two, same DNA damage also activates Peter in a p53 independent manner. Now, last few slides, I'll finish it. What is the significance of this finding? What is the finding I have? I have the non-coding RNA, long non-coding RNA Peter present in very high levels in the brain tumor cells, number one. Number two, one of the targets for Peter is TRIM28 protein coding RNA, right? So by binding, it increases stability of TRIM28 protein coding RNA. When the stability increases, the TRIM28 protein level is protein also made in large quantities. What is the function of TRIM28 protein? It's a E3 ligase similar to MDM2. It ubiquitinates p53, keeps the p53 level very low. Okay, if these are all true, if we can manipulate the Peter level in context of the tumor, it should regulate the tumor growth also, right? So the brain tumor cells, if you inject into the brain of the mouse, it will grow as a tumor. Okay, obviously one has to use nude mice, not the normal mice. Nude mice, lacks the functional immune system. So one can take human brain tumor cells which are tagged with the luciferase gene, firefly luciferase gene, and luciferase tagged brain tumor cells are injected intracranially into the mouse brain, nude mice brain, and the tumor grows inside. When the tumor grows, more the brain tumor cells, more the growth of the tumor, more luciferase will be made. When you inject luciferin, the substrate for luciferase, and then measure the amount of bioluminescence, one can measure that as a growth of the tumor. You can see on the left side, the bunch of animals, day 7 to day 25, the bioluminescence, the red color, which is a measure of bioluminescence, red to blue. Blue is a less amount of light, red is a huge amounts of light. You can see the tumor is growing bigger and bigger from day 7 to day 25. On the left side, these are control animals. On the right side, if you silence Peter, okay, if you silence Peter, tumor growth comes down. You see hardly any tumor on the right side on day 25. Why? When there is no Peter in the brain tumor cells which you are inducting into the brain, what will happen? When there is no Peter, TRIM28 RNA stability will go down. TRIM28 protein will go down. P53 protein will remain there in the cell. P53 will induce cell cycle arrest and apoptosis. Tumor growth goes down. So this is one way we prove in the absence of Peter, the tumor growth comes down. That is because P53 gets activated when you silence Peter, P53 gets activated. That brings about cell cycle arrest and apoptosis. Tumor growth comes down. You can see on the right side. Right? And in the next slide, you can see instead of silencing Peter, if you overexpress Peter, it will promote proliferation because it is going to inhibit the P53 function, right? So you can see the two sets of tumor, one on the left and right. Left side tumor is growing bigger, on the right side tumor is growing even bigger. That's because high levels of Peter is able to decrease even little bit of P53 which is present and promotes the growth. And we also show that promoting growth by the higher expression of Peter is carried out through the TRIM28 because if you simultaneously inhibit the expression of TRIM28, overexpression induced tumor growth is absent. And also we show in the next slide, the bottom portion, and another experiment, the increased tumor which is growing bigger in the presence of high levels of Peter is also resistant to chemotherapy. Okay. Final slide. Okay. This is the last slide. Okay. So what is the real benefit for a patient? If you have a bunch of glioblastoma patients today, let's say there are 100 brain tumor patients. Some tumors, P53 is mutated. I told you in the very first slide, P53 gets mutated in the cancer, right? But not all tumors are mutated. Some tumors have wild type P53, some tumors have P53 which is mutated. If the P53 is mutated, in those cells, in those tumors where P53 mutated, Peter has no function, at least with respect to regulating P53, because mutated P53 is not functional. So whether the Peter level is high or low, whether it regulates TRIM28 or not, it has no relevance in terms of growth regulation because P53 is not functional. Whereas in those cells, P53 is wild type, Peter levels can regulate the P53 function. If the Peter is wild type in a given tumor, high levels of Peter can keep the P53 inhibited all the time. If the level of Peter is low, P53 level can increase. So in a P53 wild type containing tumor, Peter levels can regulate the P53, which means thereby it can regulate the tumor growth also. So P53 wild type containing tumors, Peter levels can regulate P53, thereby can regulate the proliferation of the tumor or growth of the tumor. Which means if you take the 100 brain tumors, divide them into wild type P53 containing and mutant P53 containing. Those who are having wild type P53 containing tumors, if you divide them into high Peter and low Peter, those subset of wild type P53 containing tumors where the high levels of Peter is found, you will have a good prognosis because high levels of Peter will keep inhibiting P53, right? And that will make the tumor to grow bigger and bigger. That will lead to poor prognosis because the growth of the tumor is increasing, it is not benefiting the patients. On the other hand, the same p53 wild type tumors, if the Peter levels are lesser, the p53 level increases, that will promote apoptosis, thereby the growth of the tumor is reduced, less aggressive. The patients will perform better. You will have a good prognosis. You can see here, just the last slide, Kaplan-Meier. How many of you know about Kaplan-Meier survival curve? So we have taken 101 patients corresponding to those who carried wild type p53, divided them into two groups, blue line and red line. Red line corresponds to those who had high levels of Peter. Blue line corresponds to those who had low levels of Peter. High levels of Peter containing p53 wild type tumors, you can see the median survival is lesser. It makes a tumor very highly aggressive. Patients do not respond better to the treatment. Many of them die very early. Whereas if they have a less amount of Peter, p53 gets increased. That will decrease the aggressiveness of the tumor. It benefits the patient. The blue line represents better survival. Okay, this is one of the clinically useful information coming out of our study. So that kind of a correlation is not seen in the tumors carrying mutated p53 or in the entire cohort. So I'll stop here. This is a model, very important. Students, please watch this model slide. What we show in principle is Peter RNA, which is a nuclear RNA, it binds to TIM28 RNA, increases stability of TIM28 RNA, thereby more TIM28 protein is made. The TIM28 protein will go on to ubiquitinate p53 and degrade p53, thereby keep the p53 levels low. So that makes the cells to proliferate. That's what happens in the tumor cells. Tumor cells are able to proliferate very fast because Peter levels are very high. Okay. In the context of the brain tumor, in the normal brain cells, Peter levels are lesser. Whereas in the glioblastoma, which is a tumor cell, Peter level increases. Okay. High levels of Peter will perhaps bind to subpopulation of the TIM28 RNA, and the remaining TIM28 RNA gets degraded, and that stabilized TIM28 RNA makes more TIM28 protein that will keep the p53 under check. Upon treatment of the wild type p53 containing tumors, one thing happens, p53 gets activated. DNA damage activates p53. I told you in the introductory slide. Same time, DNA damage also activates Peter. Peter now will bind, very high levels of Peter will bind to all the TIM28 molecule and thereby increase more number of TIM28 RNA, more TIM28 protein that will simultaneously go and bind to the DNA damage activated p53 and inhibits its level, thereby creating something called incoherent feed forward loop. Incoherent feed forward loop mean that same signal DNA damage on one side activates p53, other side it activates inhibitor of p53 Peter to prevent the entire process from happening. It's called incoherent feed forward loop. The last slide, we also contribute a new method of p53 inactivation, that is Peter, through its ability to regulate TIM28, is able to inhibit p53 in addition to previously known mechanisms of p53 inactivation during brain tumor formation. With that, I will stop. Dr. Samadhi Dana is a postdoc who did most of this work. The other students who participate in this work are Bhavana, Prasasvi, and Mainak Mandal, the bottom of project assistant, and my collaborators and the funding agencies. I thank them. I'll stop here. Okay, I'm done. If there are any questions, I want questions from the students.
Thank you so much, Professor Kumaravel Somasundaram, sir.
Any questions?
Any questions from the lecture given by Professor who enlightened us on the role of Peter in cancer regulation?
How many of you understood my talk? Then ask a question. Yes. Come here. Yeah, stand up and ask question.
Good afternoon, sir. My name is Hema Malini. I have a doubt, sir. Um, you you said you classify the tumor cells into wild and mutant type.
Right.
How does that mean that do both tumor cells have any different characteristics?
No, all the tumors are glioblastoma tumors. Okay, and they sequence DNA, identify which tumor has mutation 53, which doesn't have a mutation.
Okay.
So that data set is not from my lab. So there is something called TCGA, the Cancer Genome Atlas. It's a worldwide consortium started in 2008. They have got samples from different cancers and sequenced the genome of several hundreds of samples. This data is available freely. I'm telling for all the students and teachers here. Cancer Genome Atlas, the TCGA, have sequenced several hundreds of genome of every other cancer, not only brain tumors, breast cancer, cervical cancer, lung cancer, kidney cancer, all kinds of about 33 different cancers they have sequenced. From that only we have downloaded the data. There we analyzed. Only in the p53 wild type containing tumors, the levels of Peter can regulate the survival of the patients. So that's how it is identified. They sequence the genome.
Yes, sir. Thank you, sir.
Yes. Any other questions?
Sir, good afternoon, sir. I am Jamuna. Sir, you said that in case of tumor, the P53 level increases. My doubt is, sir, tumor occurs because of the suppression of tumor suppressor gene like
Right.
P53 and the activation of oncogene.
You're right. So question from the student, what's your name?
Jamuna, sir.
Jamuna. Jamuna is asking me, tumor arises due to suppression or inactivation of tumor suppressor gene and activation of oncogenes. This is true, which means our question is P53 is already suppressed. Why do you need Peter to suppress P53? Is that the right question?
Yes, sir.
If P53 is already inactivated, why do we need one more inactivator of P53 like Peter? The function of Peter, as I explained to you, is to inhibit P53. The question here, again, I will tell you one more time. If P53 is already inactivated during the brain tumor development, why do we need another inhibitor when P53 is already inactivated? Is it understood? The answer to that is yes, P53 is inactivated during brain tumor development or any other tumor development, but not in all samples. Okay. If you take, for example, the last slide I showed you, 100 brain tumor samples, only 28% of the tumors carry mutations in P53. Is my slide still connected? It's okay, don't worry about it. So only 28% of the brain tumor samples, P53 undergoes mutation but inactivated. Remaining 72%, there are no mutations in the P53. So there are other mechanisms of P53 inactivation occurs. The contribution from this work is Peter-mediated downregulation of the expression, thereby inhibiting P53 is one more novel method by which P53 inactivation occurs. So in all those tumors where P53 is not mutated, other methods by which P53 gets inactivated, Peter encodes for one such non-genetic method of inactivation. Is it understood?
Okay, sir.
Yes.
Thank you, sir.
Any other questions?
Sir,
Yes.
I'm Mohammed Samir, sir.
Okay.
So my question is,
yes,
if we somehow, if we inhibit the Peter
yes,
protein, is there any other complication can be induced if we inhibit the Peter?
Very good question. So first of all, can we use the idea of inhibiting Peter to decrease the tumor growth? Yes, it is possible. What will happen? What is the function of Peter? Peter keeps the P53 level low, right? When the P53 level is low, high Peter, low P53, it will increase the proliferation of the tumor, correct? So if you inhibit Peter, P53 will increase, right? And that will inhibit the tumor growth. The tumor burden will come down. The patient will behave better. They will respond to the treatment. So inhibiting Peter can be in principle used as a method to inhibit the tumor growth. The question is, will it lead to any other complication? Correct.
Yes, sir.
Interestingly, it appears it may not lead to any other complication because the moment we identified Peter is expressed at high levels in the tumor, two things we did. Is a Peter level is up, elevated in any other cancer? None of the other cancer Peter level is high. It is high levels of Peter seen only in the brain tumor, number one, not in any other tumor. That's one thing. I don't know whether it's good or bad. It is present only in brain tumor at high levels. Second is more important. We looked at another database where normal tissue derived RNA is quantified and the level of different RNA, coding and non-coding RNA, levels are measured in the different normal tissue. It's called GTX platform, where expression of all the known RNAs were measured, quantified. The levels are shown. You can actually put the name of the gene. It will explain, it will show you output, the level of, let's say, P53 RNA, Peter RNA, whether it is different between the normal tissue, in the normal body. It so happens the Peter level doesn't increase in any of the normal tissue, including normal brain tissue. None of the organs, the levels are very high. Only tissue, the level is very high is testis. Okay, that is because what is the function of Peter? To inhibit P53, thereby increase the proliferation. What is happening in the testis? The sperm cells are made in millions and millions in large quantities, in large number throughout our lifetime. So the proliferation of sperm cells needs such an environment where genes like Peter level should be very high, P53 level is kept very, very low. These germ cells can proliferate in large quantities. So other than testis, none of the normal body cells contains high levels of Peter. Possibly the answer to your question is targeting Peter could be an approach. One can target specifically the brain tumor growth, except the complication of targeting the testis cells. Okay. So of course there are antisense mediated approaches are there. One can make antisense oligonucleotides to target the expression of the Peter in the brain tumor. Thereby one can inhibit brain tumor growth. Good question.
Thank you, sir.
One more question. Maybe we are running out of time. We can stop perhaps. Right? Okay. Thank you, everyone.
Thank you, sir. We now begin online oral and poster sessions 1, which poster sessions 1 screened through the projector. Now we hand over the session to Dr. Anita Margaret, Assistant Professor, and Dr. Uma Maheshwari, Assistant Professor, Department of Biotechnology.
Good afternoon. Can I present, sir? Okay, sorry. Fine.
Students, please maintain the decorum. Students, please maintain the decorum.
The first participant is Soumya. Soumya, are you ready for the presentation?
Yes, ma'am, I'm ready.
Are you audible?
Yes, ma'am.
Yeah, kindly share your slides. Kindly restrict only to the objective of your study, methodology, and your research and research
Yes.
outcomes, as well as your research results.
Okay, madam.
Starts now.
Can I start, madam?
Have 5 minutes to go.
Good afternoon, everyone. My name is Soumya, studying at that Sahyadri Science College, Directorate of Distance Education, Kuvempu University, Shankarghatta, Karnataka, and my co-worker, Ms. Spurti KR, Department of Studies in Environmental Science, Karnataka State Open University, Mysore, Karnataka, conducted a study on a pilot study on avian diversity around the stretches of Tunga River, Tirthahalli, Shimoga district, Karnataka, India. As we all knew that the birds are an exquisite life forms in the environment and it has adapted an airborne mode of life. Birds are very crucial to natural ecosystem by serving as insect pest controllers, rodent predators, scavengers, seed dispensers, and pollinators, helps in maintaining ecosystem balance and sustainability. Protecting and maintaining avifaunal diversity is crucial for preserving plant and animal species diversity. A work done by Vijayan in 1986 states that in Indian wetlands, 318 species of birds were recorded, out of which 193 species are fully dependent on wetlands. According to the study conducted by Vishwakarma and co-workers in 2014, wetlands are important bird habitats. They use it for feeding, roasting, nesting, rearing their young ones. Bird populations serve as sensitive indicators of pollution in both terrestrial and aquatic ecosystems, according to the survey done by Gaston in 1975. Such as declining population of specific species indicating habitat loss or pollution, while increased number of certain birds might signal ecosystem growth. As the avian population at different habitat depicts the community structure and importance of the landscapes, hence a study on avian species was conducted around the stretches of Tunga River in Tirthahalli Taluk of Shimoga district, Karnataka, India. The picture shows our study site in Tirthahalli Taluk of Shimoga district, Karnataka, which is geographically located approximately from 13° north to 75° east latitude and longitude. The area covers 8.86 kilometers square with richest biodiversity forest area surrounding. It has both terrestrial and aquatic ecosystem and visited twice in a day, and birds were counted through walking and scanning around the bridges from morning 6:30 to 10 a.m. and 4 to 6:30 p.m. in the evening from February 2025 to July 2025, which is winter and spring seasons. And as per the survey conducted by Basavarajappa in 2006, who is a professor in the University of Mysore, and Shruti and Basavarajappa in 2016, we incorporated point count method and direct count method in our studies for counting. The birds counting and photography, we used a pair of Olympus 10 by 50 binoculars, Nikon D500 camera, and Nikon AF-S DX Nikko 55 to 300 mm f bar 4.5 to 5.6 zoom lens were used. According to field guides explained and published by Ali in 2002 and Grimmett and co-workers in 2011, the bird species were identified. These are all the bird species observed in the study site where the numbers are in pairs. Altogether, 12 bird species were identified, in which Glossius philippinus is more in numbers, found in 6 pairs. The pie chart shows the distribution of bird species, where the largest slice is occupied by Glossius philippinus. These are all the photographs shot by me and my co-worker, Miss Spooty Kyar, and shot by Nikon D500 camera. That brings me to the end of my presentation. The result shows that the area is moderately rich and diversified avifauna with 12 bird species. In recent days, avifaunal diversity has been decreasing due to human disturbances, which leads to the destruction of their habitat. Research has shown a decline in bird population across ecosystems due to anthropogenic interference, including habitat loss, degradation, hunting, pollution, invasive species, and diseases. Birds aid pollination, which is critically important to ensure the production of fruit, seeds, and vegetables essential for human and animal diet. Therefore, further studies on avifaunal diversity, studies of Tunga River Tirthahalli are needed for conservation. These are all the references helped us in the study. A special thanks to Dr. J.S. Chandrasekhar, who is working as a professor and chairman from Department of Studies in Environmental Science, Karnataka State Open University, Mysore, Karnataka. Thank you.
Yeah. Let me ask questions.
Yes, sir.
Ready, ma? Soumya?
Yes, sir. Good afternoon, sir.
I would like to ask you two questions. Number one,
Yes, sir.
because you have done avian diversity in the particular location, pilot
Yes, sir.
study, right?
Yes.
Can you name some of the water birds and the terrestrial birds?
Water birds and terrestrial birds, sir.
Yeah.
So water birds, swan maybe.
Okay.
Swan and ducks, geese and herons.
Okay. Yeah. Name some terrestrial birds.
So terrestrial birds in our study, we found more Plausius philippinus and other birds, sir.
Right. Okay, ma. Another question.
Yes, sir.
Can you tell me the advanced software for the diversity indices? Did you use any software?
No, sir. Because it's a pilot study.
Okay.
The work was done for 4 months, and we will work on it further in future, sir.
Then how do you classify the birds?
Classification of birds, sir.
Yeah.
Sir, we can classify it as
Which kind of references you have gone through
sorry,
for
sir.
identification of the birds?
Sir, Dr. J.S. Chandrasekhar, a professor who helped us in the identification, sir.
You have any manual for that?
No, sir.
Okay, ma. Ma, you are very much interested in avian diversity. I would like to tell you one software,
Yes,
Merlin, Merlin. Merlin. Actually, it's an advanced software
Merlin.
for identification of the birds, right? So here you see there are three types of identifications. One is call of the bird. Okay, you can download in your mobile phone.
Yes, sir.
Any bird's call, the app detect the bird's call and it is shown in the screen.
Okay, sir.
Then photograph. If you take the photograph, then you can match with this app that will tell
Yes,
you
sir.
that which bird is this.
Okay, sir. Thank you for your information,
Okay.
sir. Thank you so much.
Yeah, thank you, ma. Just a minute.
Thank you, sir.
That was an interesting presentation and was very colorful also. We also came to know different types of birds. So what is your futuristic perspectives regarding the pandemic issues? Are you trying to create an alarm for the birds Are you all are you planning any strategies to protect them?
Ma'am, nowadays, due to anthropogenic interferences and pollution, the bird population is decreasing, which is, as I earlier said, this pollination is more important for production of fruit, seeds, and vegetables, uh, for for all the food chain and human animal diet. It helps. So now it's a pilot study. I would work on it in future, with uh, any government or non-governmental uh, uh, non-governmental fund us to work on it more, madam.
Soumya, nice presentation, ma.
Thank you.
Is there any difference between before Corona period, after Corona period, about the bird species? Have you seen?
Yes, ma'am. During Corona period.
Area?
Yes, ma'am. During Corona period, the birds were more in numbers because the people are not going out uh, and poaching and those works are less in the Corona period. So after that, it will it the numbers are decreased after that, madam. After the period, Corona period.
Are there any species extinct?
Extinct, for now, I'm not aware of this, madam. I will work on it.
Okay, okay.
Thank you, madam.
Thank you, Soumya. Moving on to next
Thank you.
participant. Ala Kayali. Are you there, ma?
Yes, ma'am. Yes, ma'am. Yes.
You are from SRM Institute of Science and Technology, Chennai. Can
Yes,
you please
ma'am.
share
Yes,
your
ma'am.
screen?
Have sharing. Ma'am, you have to allow me. So, can you allow me, please, to share screen?
We don't have the screen on. Soumya, can you please close the screen?
Yes, ma'am. I have closed. I've stopped sharing.
You can share, ma.
Good afternoon. Good afternoon, all. So I am Ala Kayali. I'm hearing my voice twice. I am Ala Kayali from SRM, Institute of Science and Technology, Department of Biotechnology. So today I'm here to present the exploring. Sorry. Yeah, today I'm here to present about the exploring the role of endophytic bacteria isolated from Helianthus annus, grown in phytoradiation enhancement. So with the guidance of Dr. Infant Santosh. So we'll start from endophytic bacteria. Endophytic bacteria is the bacteria we can isolate it from in the internal tissues of the plant. So we can isolate it from leaf, stem, and root, flower also. So it has some functions for the plant, like it can protect plants from pathogens and pests by producing antimicrobial compounds and competing with pathogens for resources and stimulating plant defense mechanisms. and it helps plants tolerate stress such as salinity, drought, and heavy metal toxicity. So it can be found in medicinal plant tissues that will give strength to the medicinal plant itself, and it's promote plant growth by improving nutrient uptake and producing plant growth hormones like auxin and cytokinin, and increasing the availability of essential minerals. So some applications of endophytic bacteria, it's like used as biofertilizers and ameliorating agents, and also in bioremediation. So why we use Helianthus annus? Helianthus annus is a herbaceous plant, so it has a short life cycle. It's like 3 to 3.5 months, and it's also worldwide grow, distributed, and it's also like the most important thing that is a hyperaccumulation plant. So it has the capacity to accumulate heavy metals in its tissues, like by phyto extraction technique. And it has high economic importance for its oil. This also can be used for biodiesel and livestock. So the aim of the study, isolation and identification of endophytic bacteria from Helianthus annus grown in heavy metal contaminated soil. So we already contaminate the soil with zinc and lead and chromium. In addition to detecting the potential role of the isolated endophytes in enhancing phytoremediation. So the methodology, after growing the plant in the already contaminated heavy soil with heavy metals, in the flowering stage of the plant, it was like 60 days. We did the surface sterilization for the leaf and stem and root, and we isolate endophytic bacteria from leaf, stem, root. And after that, we got unknown endophytic bacteria. After that, we did purification and morphological identification, biochemical tests, plant growth promoter tests, and genetical characterization. So in morphological identification, we study the morphology of colonies, like the margin and elevation. And for biochemical tests, we studied the gram staining test and endospore staining test, and also amylase, catalase, and MR and VP test. And for plant growth promoter, we study auxin production test and hydrogen cyanide production test, also nitrogen fixation and phosphate solubilization. And for genetical characterization, we isolate the DNA. After that, we did that 16 rRNA to identify the bacterial strain that we isolated. So in plant growth promoter tests, we got like some it give positive and some it give negative features. So those 5 species, they give positive in the all 4 plant growth promoting tests. So for that purpose, we take it for the genetical characterization, and we got the results, that is Bacillus anthracis, Bacillus tropicus, and Providencia vermicola, and Bacillus thuringiensis, Bacillus cereus. And those 5 species, by searching in literature review, all the 5 species were used in bioremediation. So already they used to enhance the bioremediation to remediate the heavy metals. And this is like we did the phylogenetic tree by Mega 10 software. So the conclusion, the isolated endophytic bacteria not only contribute to plant growth, but they may enhance the performance of hyperaccumulator plants in the phytoremediation process due to their proven capability in bioremediation. Future research prospects include investigating the application of isolated endophytes on the other hyperaccumulator plants and verifying the released secondary metabolites and searching for the mechanism which this bacteria enhance the phytoremediation. Those are some references used in this study. Thank you. Any question? Hello, ma'am. Hello.
Ala Kylie,
Yes, ma'am.
you have worked in endophytic bacteria, isn't it?
Yes, ma'am.
Okay.
The bacteria.
How are you saying that these bacteria are playing role in phytoremediation?
Yes, ma'am. Already this bacteria were used, were applied in the previous studies in bioremediation. So it enhanced the bioremediation. It's used for bioremediation to remediate the heavy metals. So it has that heavy metal resistant activity. Like already studied, ma'am, in previous studies. So just we did the characterization. And after characterization, we know that those species already used in bioremediation.
But you need to prove some experiment, isn't it? You have just done the isolation. According to my understanding, you have just done the isolation, and you have did you progress the 16S RNA sequencing, and did you publish it in NCBI?
It's still not published, but I did morphological identification by chemical test, plant growth promoter test, and then genetical characterization also. We didn't submit it to NCBI till now. We will submit.
In that case, what should be your futuristic outcome?
Ma'am, we can apply those isolated endophytes on another plant to enhance the phytoremediation also, like for another plant.
Okay.
Mhm.
Okay, ma. Thank you so much.
Okay,
The
ma'am.
next
Thank you.
participant, Dr. Monica Chaudhary, Banzai Lal University. Dr. Monica Jangra, are you there? So in that case, we'll move on to the next participant, Dr. Seema Rani. Dr. Seema Rani. Okay, Spurti KR from Karnataka State Open University. Can you share your slides, ma? Spurti, are you there?
Ma'am.
Hello. Hello, ma'am.
Hello.
This side, Dr. Monica Jangra. I am audible?
Yes, you can start. Focus on your objectives, aim of the work, your methodology, results, and results outcome.
Is I'm audible?
Yes, Purti, you can. You can start your presentation. We can
Okay,
hear
ma'am.
you.
Okay. Good afternoon, all. Myself, Spurti Kar, MSc student from Karnataka State Open University, Mysore. And my topic of presentation is about assessment of chlorpyrifos 50 EC insecticide against the control of white stem borer Xylotrochus quadripus in coffee plantation around Chikmagaluru district of Karnataka, India. Assisted by Dr. Chandrasekhar, Professor and Chairman of Department of Studies in Environmental Science, Karnataka State Open University, Mysore. As we know that Chikmagaluru in Karnataka is one of the major producer of coffee in India. Coffee is not only a commercial crop but also a livelihood for thousands of farmers in Western Ghats. However, one of the major threats of coffee cultivation, especially coffee arabica variety, is white stem borer. From the study of Reddy and his co-workers from 2019, it was found that white stem borer is the most damaging pest responsible for causing yield loss ranging from 35 to 90% in coffee plantation across India. The white stem borer is a serious pest of Arabica coffee. The adult beetle lays eggs in bark and larvae bore into the stem. This
Kindly go to materials and method, ma.
The image shown here is the Chlorophyllus 50 EC insecticide, which is widely used as insecticide to prevent that. This belongs to organophosphate group and is considered to be a broad spectrum insecticide, which means it controls wide range of insecticides. This group of insecticides work by inhibiting an important enzyme called acetylcholine esterase. This enzyme need for the proper nerve function in insect. When it is blocked, the nervous system gets overstimulated and cause paralysis and eventually death. It means it finally leads to neurotoxicity in insect. The formulation which is shown here is 50% emulsifiable concentrate. Farmers generally use it for foliar spray and stem application. That's why Chloropyrifos 50 EC is effective because it targets the insect nervous system and widely used in pest management. Study area I have taken. Study was conducted in Chikmagaluru district, which is located in geographically located between 12 degree and 13-degree north to 75 and 76 east longitude, situated in the Malnad region, known for its favorable coffee growing state. Methodology. The study was conducted in some selected coffee plantation in Chikmagaluru district of Karnataka from March to May 2025. In this experiment, we used Chloropyrifos 50 EC and it was directly sprayed on the coffee plant stem. The aim was to target the white stem borer, particularly larva and eggs. We monitored several aspects after spraying, like changes in the borer population dynamics and the health and growth parameters of the plants, like if the plants is recovered or grow better, like that, and any possible phytotoxic symptoms, like any side effect of the chemical on that coffee plant. Uh, um, we maintained standard methods and protocols of Reddy and his co-workers, like periodic field observation and sampling were collected for both effective and adverse effect of the treatment of Chloropyrifos 50 EC. This method helped us to understand not only how effective this insecticide was against the white stem borer, but also whether it had any side effect on the coffee plant. Result: direct stem application of Chloropyrifos 50 EC effectively targeted neonat larvae, which means young larvae and eggs. Young larvae and eggs of the quadrupus. So controlling them early is important to prevent the damage. As I told earlier, this insecticide act as a neurotoxin, as something that affects the nerve cells, stopping the insect from feeding and moving properly. Post-treatment monitoring showed the significant decline in the white stem population across the treated coffee plantation. Research demonstrating that chloropyrifos 50 EC act as a both ovicidal and larvicidal efficiency. When compared to other pest management, it shows high effectivity. But the residual effect to environment is varied depending on several factors such as photo degradation application method. Discussion part from the from the result, we have concluded that the chloropyrifos 50 EC provide effective control when applied in the early pest stages. But this chemical act as a dual edge, which means it has both positive and negative impact. It also impact on the pest controlling, but and also it has a negative impact on plant physiology, deprived cellular structure and antioxidant response and photosynthesis also, which means if the farmer use in a higher dosage, it also affect the main crop also. These are the conclusions that I have made. Chloropyrifos 50 is effectively managed white stem borer, which has a significant contribution to pest population reduction. The insecticide act as a neurotoxin targeting neonate larvae and eggs. But some few negative impacts also associated with the higher dosage of the insecticide. These findings highlights the need for further research to sustainable and integrated pest management strategies and need to be managed, control effectiveness with environmental safety and plant health. These are the reference. Thank you.
Yeah, thank you, Sproti. Actually, you have done.
Yes, sir.
Actually, you have used the chemical pesticides, insecticide.
Yes,
okay, it is a chemical method. Okay,
yes,
right.
Sir.
Did you find any alternative methods for biological control of white stem borer? Did you find any alternative way?
Biological method is cutting the stem in early stage.
Okay.
And usually these white stem borer are favorable to shady regions. So avoid too much shadiness and those types of cutting and shade region should be avoided.
Now, what I'm asking, instead of using this insecticide which you have mentioned,
yes, sir.
Is there any natural enemy for the white stem borer? Why can't you encourage that to control this white stem borer instead of using insecticide? Did you find any enemy species of white stem borer in the field?
Yes, sir. Some few birds are there.
Can you name them?
Sorry, sir. I can't get it.
Okay, ma. Thank you. Thank you.
Thank you.
Thank you, ma. Thank you, Spurti. So next is Darshan Kumar T, University College of Science, Department of Zoology.
Yes, ma'am, I am available. Yes,
Can
ma'am,
you
I am
please
available.
share your
Yes,
slides,
yes,
sir?
yes, ma'am, I am sharing now. दोबारा message. Is it my PPT showing, madam?
What happened? Darshan? Yeah. Can you share your screen?
Madam, it is not sharing properly. I'm trying to share. It is sharing, madam. Is it sharing, madam?
Your file is not opening.
Is it sharing, madam? Now it is open.
Yeah, we can see. You can start. You have 5
Oh,
minutes. Concentrate
okay.
your objectives, results, methodology, and future outcomes.
Okay, madam. Okay. My name is Darshan Kumarti. I'm a research scholar doing PhD in the Department of Studies and Research in Zoology from the University College of Science, Tumkur University, Tumkuru. Under the guidance of Dr. Parimalabi, is working as Associate Professor in the Department of Zoology, University College of Science, Tumkur University, Tumkuru. So my research topic is on halosulfuron methyl induced determination of LC50 with behavioral studies in the freshwater fish, Cyprinus scorpio lineus. So halosulfuron methyl, it is a globally used as a sulfonylurea herbicide. It is extensively used in the field of rice and sugarcane in the Karnataka because application of halosulfuron methyl, it is used in the low doses. It causes, particularly, it helps in eradicating the weeds in the particular in the rice field as well as in the sugarcane field. So as well as by incorporating of this halosulfuron methyl herbicide, it causes also effect to a particular type of an environmental as well as aquatic effects, and aquatic organisms, especially fishes, are vulnerable to pesticide. As we know that if it sprays to a particular type of an environment, it can also be goes to a runoff, and it causes damages to aquatic life forms. So as well as their residues also called as a neurotoxic and physiological disturbances, and it is also shows that there is a growing evidences in selective herbicides like halosulfuron methyl can also cause toxicological and behavioral changes was observed. So methodology involves the LC50 determination. Goes to the OECD guidelines, which is OECD means Organization for Economic Cooperation and Developmental Guidelines, according to the test number 201. So acute toxicity assessments were performed. According to, I taken a 10 group of fishes of Cyprinus scorpio. These fishes were exposed to halosulfuron methyl for a period of 96 hours, and in the 96 hours I monitored completely what are the changes have been occurred, that is the behavioral changes, as well as any of the fish species is getting to be mortalities is observed. So I observed, and also it is also calculated in a statistical manner that the LC50 value is observed in a statistical manner that will be about LC50 of fish population was expected to die within 96 hours of exposure period, and I found that the median LC50 concentration was 557 mg per liter for the halosulfuron methyl herbicide. So this is what is the results is shown here. This is from the serial number 1 to 9. The concentration is maintained in the milligram per liter. I also maintained the log concentration, which will be statistic, which is be helpful in the statistical analysis, as well as the number of fish exposed in every 96 hours is 10 fish species, number of fishes alive, the number of fishes dead, and percent kill and a probit kill. So log concentration, it is a logarithmic transformation of the concentration facilitating analysis of dose response relationship. Percentage kill, it indicates the lethality of the herbicide, where I mentioned that the 50% which is marked in a yellow color in a chart that shows the LC50 value. That is LC50 value means 50% kill of a particular species of fish. So probit kill, it is a statistical measure used to analyze the mortality data, helping to estimate the relationship between a dose and the responses. So this is the analysis plot. In the statistical manner, I calculated R square value which will be lies between 0.9909. R square value 0.9909 is extremely high, that is very close to 1, which will be indicating a very strong and positive linear relationship between the log concentration and the probit kill, and this suggests the experimental data is very reliable. This is the plot plotted between the probit kill as well as the log concentration. The log concentration is measured in the mg per liter, which will be converted to the log concentration. This is the percent mortality sigmoid curve. So as the concentration, as we observe here, the percent kill as well as the log concentration, so there will be a significant S-tape sigmoid curve is observed where the R square value is 0.9943. where observed that the concentration increases, there is a mortality or the killing percentage is also increases with respect to the particular mortality sigmoid curve. At low concentration, mortality is low. As the concentration increases, mortality rises. At a very high concentration, mortality near to the 100% as I shown. This curve represents the full range of substances of toxic effects. So this is all about the LC50 value. Now I am moving on to the behavioral responses in Cyprina scorpio. I observed several behaviors as lateral swimming, sinking phenomenon, all those things, and which will be observed for the 96 hours. I also kept in another aquarium in the control environment, and another aquarium I exposed to halosulfuron methyl for a 96 hours, 2 hours exposure, 24 hours exposure, 48 hours, 72 hours, 96 hours. So consistently I recorded the different types of what are the happenings in the physiological behaviors responses. So it shows that there is a behavioral changes such as swimming abnormalities, loss of equilibrium, and respiratory distress were observed. And as well as this is the video, just a 10 seconds video. This shows that this shows that there is a wing, there is a reverse swimming as well as the upward swimming as well as the back swimming, and it is also causes a loss of equilibrium behaviors. It is swirling from 2 to 3 times in a particular type of an aquarium. So this is the what happens for a fish species. It will mainly it causes upward swimming, vertical swimming, loss equilibrium behaviors, and several additional behaviors also was studied. So this is the determined acute toxicity of halosulfuron methyl was observed is 557 mg per liter in the Cyprina scorpio species. Herbicide exposure can cause a dose-dependent abnormal behavior responses, indicating that neuromuscular and respiratory distress. Because all these things, all these behaviors will be caused because in the nervous system there is a acetylcholine enzyme activity will be distressed. So because of all these things, the particularly fish species is going to be prone for the showing that the several different types of behaviors, and there will be behavioral changes included, that is lateral swimming as well as lots of equilibrium and schooling behavior, which means the fishes will move in a group, that is called as schooling behavior, and fight responses, and burst swimming because of sodium and potassium channels activation. It is causes a burst swimming, and it is also opens for excessive opercular movements, and all these behavioral studies was observed. So these are all my references. So thank you.
Thank you, Darshan.
Yes, sir.
I have a question. How many samples you have taken for this study? And then, did you find any mutational behavior when you applied for LC50?
Sir, mutational behavior, that is the further objectives I have taken, sir. So first I taken as halosulfuron methyl, which will be primary calculative for LC50 value, and in this LC50 value I was exposed for the 96 hours. So I will expose it for further to in case of 40 days to 60 days for the continuous exposure. What are the different types of organ failure or histopathological studies has to be determined, that it will be involved in the objective studies, as well as I will also have a plan to do a particular type of genotoxicological studies in the further in our PhD results, sir. So I need to do it in a very strict manner, so that this is the first objective which has completed, and all those things what I shown in the PPT, those are all the evidences, sir.
Have you gone through the
yes, sir.
Genome of this fish? Genome study?
No, sir.
Earlier,
I need to do it, sir. Genome studies. So that's my next objective to study. So I will mention some protocols. That means MN nuclear studies and those things. I need to do it, sir. I am not done in this. But the main objective of this study, in the first objective, is that to determine the LC50 as well as Cyprina scorpio behavioral study. What is why the exposure of 96 hours of halosulfuron methyl herbicide, sir.
okay, thank you, thank you.
Okay, sir. Okay.
Darshan, we can see the realistic work that you have
Yes,
put on, and
yes,
that
sir.
video was really fascinating.
Okay, ma'am. Thank
My
you.
question is that, like,
Yes,
when was that behavioral study in accordance to the median LC50 value that you have reported, which was
yes, ma'am.
around 500 and something?
Yes, ma'am. That's the value for LC50 value, madam. While exposed, I think so that there will be an where while I go for Google's scholars and I've read some research papers, it shows that they clearly indicate that there is a high effect on the nervous system because the acetylcholine stress activity will be limited and it will cause some distress to a particular type of fish species. So I need to do further work. So this is my first objective completed, madam. So which was explained in this conference, madam.
What sort
Detail.
of neurotoxicity assay are you planning in your futuristic? Because you are doing on behavioral, isn't it? So are
Yes,
you
ma'am.
in the plan to do any neurotoxicity assay?
Yes,
For
ma'am.
example, tomography study of the brain or something like that?
Yes, ma'am. That's what I have planned to do. That, ma'am. That is histopathological studies of brain as well as gills and nervous system, as well as the liver and all those things, which has to be exposed from the 96 hours and which will be calculated. I have taken two objectives in that means that is lethal concentration, which will be in a low concentration. I also induce the high concentration of a particular type of species. So then I can correlate with these two studies, and I will write a thesis on these things, madam. So this is my
What was
for
the
future studies.
dosage? What was the mode of the dosage? Was it an oral or did you put it in the water?
No, ma'am. I put it in water. Put it in water, which will be as per OECD guideline. That is the Organization for Economic Cooperation and Development, which is a test number 201 is followed, madam. As per standard procedure and protocols, I followed this methodology, madam.
A kind suggestion from my side, since I'm
Yes,
also
ma'am.
working on zebra fish.
Okay,
Like,
ma'am. Okay.
since your animal is a bigger one and the size is more, so try to
Yes,
go
ma'am.
for oral dosage in future and try to
Okay.
connect your gut and your brain
Okay.
also, because many studies are being dealt with gut brain axis.
Yes,
So
ma'am.
try to do that, and also coordinate your weight of the fish whenever you're
Okay.
doing your LC50 studies. When you
Yes,
are
ma'am.
plotting a probit analysis, try to
Yes, ma'am.
correlate the weight of the fish
Okay, ma'am.
and uh, in accordance to your behavior also. That was a great work, Darshan. Thank you.
Okay, ma'am. Okay. Thank you, ma'am. Thank you for your suggestion, madam. Yes, yes, sir. Next.
Darshan, your work is nice. Have you
Yes, ma'am.
seen any specific activity for a psychological disorder like anxiety or depression?
Yes, ma'am. What I explained, there is a bird swimming and all those behaviors. What I explained there now, madam. So when fish moves slowly and it will burstly swim in a faster movement. So I observed in some research papers and where the work is going on, madam. I need to look into these parameters also for consideration in the future, madam. I was looking also under for this also, madam.
Okay, okay. It's having a high scope. Okay. Thank you.
Yes, ma'am. Okay. Thank you so much, madam.
Thank you. The next
Okay.
participant is Tania Sagar. Tania Sagar, are you there? University of Jammu.
Yes, ma'am. I am.
Kindly share your screen, ma. Kindly minimize your
Ma'am, not able
introduction.
to show.
Kindly minimize your introduction and straightly go into your methodology. Focus more on your results and conclusions.
Okay, ma'am. Good afternoon, everyone. Myself, Tanya Sagar, PhD Research Scholar from Biotechnology, University of Jammu. The title of my presentation is Comparative Analysis of Phytochemicals in Amaranthus cordatus
Tanya,
collected from different
your
regions.
screen is not visible, ma.
Now,
You have any problem? Yeah, okay. Yeah, we can see your screen now.
okay, ma'am. Good afternoon, everyone. Myself, Tanya Sagar, PhD Research Scholar from School of Biotechnology, University of Jammu. The title of my presentation is Comparative Analysis of Phytochemicals in Amaranthus cordatus collected from different regions of Jammu and Kashmir. Coming to the introduction part, Amaranthus cordatus, a multifunctional pseudomiller, belongs to Amaranthaceae family.
Can we see your methodology? We understood. Can
Yes,
you
ma'am.
please do the methodology?
Coming to the methodology, firstly, the sample collection was done for both leaves as well as seeds, which was followed by the preparation of extracts. Then preliminary phytochemical and quantitative phytochemical analysis was done. The plant material was collected from 9 different areas of Jammu and Kashmir, which includes 5 districts, that is Jammu, Anantnag, Poonch, Riyasi, and Rajouri. After that, the qualitative phytochemical analysis was done for both leaves as well as seeds of Amaranthus cordatus for different accessions, and we observed the key metabolites present in variable amount in all the accessions. Then the quantitative phytochemical analysis was done, and for this we studied total phenolic, flavonoid, tannin, and saponin content. For the total phenolic content, the highest content was observed in the accession that was collected from the Poonch district, whereas the lowest was observed in the accession collected from Riyasi district of Jammu and Kashmir. While in seeds, the highest was observed in the Jammu district accession, while the lowest was observed in the Poonch district accession. Similarly, total flavonoid content was analyzed, and what we observed in the leaf accessions, the highest content observed in the Riyasi district accession and the lowest observed in the Poonch district accession. Whereas in seeds, the variable content was observed and it ranged from 3.02 to 0.223 mg per gram. Then total tannin content was also analyzed, and we observed the highest content in leaves reported in the Jammu district accession, whereas the lowest was observed in the Rajouri district accession of Jammu and Kashmir. While in seeds, the highest content observed in the Anantnag district accession and the lowest observed in the Surankot area of the Poonch district accession. Similarly, total saponin content was also analyzed, and we observed the variable content in both leaves as well as seeds. And from this, we concluded the very key metabolites were present in all the accessions and in different quantity. So future prospects, the significant variation in phytochemicals among accessions suggests potential for selecting region-specific varieties of Amaranthus cordatus for use in functional foods, nutraceuticals, and fortified products. Also, the accessions with higher level of these compounds can serve as elite parents in breeding programs, aid and enhancing nutritional and stress resilience traits in future cultivars. And the future study should evaluate toxicological profile, particularly of saponins and tannins, to ensure safety for human consumption and identify safe threshold or required processing step to reduce anti-nutritional factors. These are the references that I have used. Thank you.
Matania.
Hello.
Hello.
Yeah, Tanya, are you here?
Yes, sir.
Okay, ma. Your topic is you have collected the plants from different regions of Jammu and Kashmir.
Yes,
I
sir.
just want to know, you have collected the plants from different elevation level or in the same plain area?
Different elevation levels, sir.
Right, okay. Then, did you find any comparative, okay, or a connection with the soil and your plant? What are the components you find in a sample? The components are same where you
Sir,
have collected.
I find diversity in all the accessions means their quantities. I find the difference in the quantity in all the accessions. That is both for the leaves as well as also for the seeds.
Yeah, that I understood, ma'am. No. See, when the soil type is different,
yes, sir.
Definitely the plant components also might be changing, you know. Did you find any difference? Because you have compared, yeah. Did you find any different phytochemicals in the different
Sir, you are
levels?
not audible.
Right, ma. Okay, thank you.
Yes, sir. I find it different.
Thank you. Moving on to the next participant. Haripriya, JSS Academy of Higher Education, Ooty.
Yes, ma'am. I'm here, ma'am.
Kindly share your slides, ma'am.
Yeah, okay, ma'am. Ma'am.
Right, right. இது மட்டும் தான் 10 right, okay, okay. விட்டுருங்க, publish okay, madam.
Good afternoon to everyone, respected chairperson, esteemed panel members, and fellow participants. And I'm Haripriya, PhD scholar in the Department of Biotechnology at JSS Academy of Higher Education and Research, Ooty campus. As a part of my MSc dissertation, I carried out a project work entitled as Formulation and Development of Bioactive Ointment for Psoriasis Management using Cleome viscosa lin seeds.
Haripriya, we can't see your slide, ma. Share your slide.
When can we start?
Yes, please.
Yes, ma'am. Yes, this work was carried during my MSc dissertation work. And basically, psoriasis is a persistent skin disease, and it has no permanent cure. It affects the quality of life of the patients. So I have chosen a plant-based alternative for this psoriasis management. And I use a plant called Cleome viscosa lin, which is commonly known as dog mustard or wild mustard. This is the methodology I have adapted, where I collected the plant material in and around Metupalayam, and I authenticated in Ooty in the Arts College. And extraction was done by using NXane. NXane, why? Because NXane, it is a non-polar, it separates out the oil because of its non-polar solvent. Basically, it is a non-polar solvent. So that is why I use this NXane solvent. And GCMS analysis was carried out. And cytotoxicity analysis using MTT assay was done using A375 cell line, which is basically known as human melanoma cell line. And final product formulation and evaluation was done. And this is the results where from extraction I have got 0.12% yield, which is a low yield. This is one of the challenges I faced during my research work. And in GCMS analysis, 13 compounds were present, and of that, linalool is the major peak obtained with a retention time of 28.57. And linalool has the properties of antimicrobial and skin soothing properties. Maybe this compound is responsible for managing the psoriasis symptoms. And cytotoxicity assay was carried out. And until this μg per ml, there was no toxic effects found when the sample was injected. And formulation and development of ointment was done, and various formulation was carried out. This is a basic formulation of ointment which I have done. And the parameters was evaluated, which with the color of white, and odor is pleasant, texture is smooth, and greasiness and it is greasy, and it is a homogeneous with a pH of 5.4, which is the range for which is applicable for the topical application. And it is washable. And finally, we have extracted the plant material, extracted the seeds from the plant material, and ointment was formulated. And in my future, I conclude that this is an affordable and safe impact, reduces dependency on costly synthetic drugs with side effects, and it improves patients' quality of life, because the patients with psoriasis, they are not satisfied with their looks, and they feel like shame, guilt, or flawed self perception. And basically, psoriasis is a visible nature of the condition, is a major contributor for this impact. So, I have thought to do this project, which is a plant based alternative for psoriasis with no side effects. And this study is yet to take to
Haripriya,
yes, ma'am.
Haripriya, in which part you have included psoriasis test, ma? You have seen your MTT assay in melanoma cancer cell only, no?
Yes, ma'am.
How are you saying it is effective against psoriasis?
Ma'am, I just tested with the whether my extract is toxic or not, ma'am. For that, I have done this MTT assay. And for psoriasis, we are planning to do in our future studies, ma'am.
Then why you have put the formulation of bioactive compound for psoriasis management? You didn't any work related to psoriasis, isn't it?
Ma'am, with the available cell line, I did my work, ma'am.
In future, try to quote the authentication that you have done. You said that you have done the authentication of the plant. They would
Yes,
have
ma'am.
given you a number. So try
Yes,
to
ma'am.
quote that number in your presentation, and then we need to know what type of cell strain did you choose, the source of your cell strain. It's not there actually. So try to focus on those aspects, ma.
Yeah,
All
sure,
the best
ma'am.
for your futuristic work. Thank you.
Thank you so much, ma'am.
Kavya. Kavya, are you ready, ma? From JSS Academy of Higher Education.
Yes, ma'am. I'll share my
Please. We need your methodologies precise, your results, and your futuristic outcomes.
Ma'am, I'm not able to share my screen, ma'am.
In that case, I'll go to the next participant. Mamta.
Ma'am, it's showing like host disable the participant screen sharing.
Haripriya has closed the screen, ma. You can share now. Do you want time? I'll move on to the next participant and then you can proceed.
Ma'am, you have disabled the screen sharing, ma'am.
Now is it okay? Try sharing now.
Yes, ma'am.
Yes, we can see.
Shall I start, ma'am?
Yes, yes, ma'am.
Good afternoon, everyone. Myself, Kavya, research scholar in the Department of Biotechnology, JSS Academy of Higher Education and Research. And I'm going to present my MSc project entitled Formulation and Evaluation of Herbal Sunscreen Gel from Trigonella phonum gracium and Punica granatum peel extract using microencapsulation technology. Introduction. Our body's external surface is being covered by the skin. So exposure to UV radiation causes various skin diseases. So sunscreen protect the skin against
Please
the...
go to the methodology, ma.
Okay, ma'am. And methodology includes the extraction of the fenugreek seed mucilage and the extraction of pomegranate peel and phytochemical analysis of these two extracts and microencapsulation of these two extracts and formulation of these sunscreen using these two extract microbeads, and finally the analysis of physiochemical parameters, antioxidant properties and SPF of the formulated sunscreen gel. And for the mucilage extraction, the fenugreek was soaked in distilled water and it was boiled at 80 degrees Celsius, and the mucilage was separated using masking cloth, and it was precipitated using acetone, and the precipitate was dried and powdered, and pomegranate extraction was done using macration method, and these two extracts were subjected to the phytochemical analysis, and a phytochemical study of these two extracts contain the alkaloids, phenols, flavonoids, saponin, steroid, tannin, and glycoside compound as active phytoconstituents. And these compounds contribute to the antioxidant activity of the extracts. So these extracts are microencapsulated using inotropic gelation technique using sodium alginate and calcium chloride, and sunscreen gel was formulated using containing these microbeads, and results and physical characteristics of the gel. The appearance of the sunscreen was gel-like. The color was transparent. Consistency was good, and it feels non-sticky. And chemical characteristics of the gel: the pH was 6.3, viscosity 11728 centipores, meeting the sunscreen parameters. The spreadability was good. The stability study of the gel was checked. The gel was kept in different room temperatures, and there was no change in the appearance, consistency, and feel. And antioxidant activity of the gel was checked, and the results showed that IC50 value was 37.56, and it was efficient for the photoprotective. And the sun protection factor was calculated using a formula, and we found the SPF value of 30. It means that it can block about 90% of the UVB radiation from the sun. So conclusion.
So formulated sunscreen gel along with the microplastic micro encapsulation technology present a promising alternative conventional sunscreen containing microplastic. The developed sunscreen formulation offers protection against UV radiation attributed to the natural photoprotective properties of the extract. Moreover, the micro encapsulation enhances the stability and efficacy of active component, prolonging the sunscreen effectiveness upon application. Thank you.
Kavya, did you find any sunscreen which is actually incorporated with microplastic?
Yes, ma'am. Most of these sunscreen, not only sunscreen, ma'am, many cosmetic products are incorporated with microplastics, ma'am. So because of these microplastic, we have so much negative health effects, ma'am.
Okay, so that was your objective. What is the real objective of your study now?
Ma'am, I'm not able to hear, ma'am.
What is your objective of the study, exact objective of your study?
For replacement for the microplastic beads, ma'am.
So in that case, what is the novelty, what is the alternative that your formulation has?
So natural microbeads, ma'am, using micro encapsulation technology, ma'am.
Okay, so what is the purpose of those microbeads? Why the formulation should be in a bead formation?
Ma'am, it is more effective than the other formulations, ma'am.
Okay, in that case, I'm coming to the same question. In that case, how do you substantiate with the microplastic?
What, ma'am?
How do you substantiate your microbeads with microplastics? How do you justify yourself? I appreciate your work, but then try to focus on your title as well as the work which you are doing now. Okay?
Okay, ma'am. Okay. Thank you.
Like in that case, like you have to focus on the need, okay, the problem first, and you are finding a solution, isn't it? In that case, you need to justify yourself. So that's what we are trying to explain from our side.
Okay, ma'am.
Great work. All the best for your futuristic attempts. Thank you.
Thank you, ma'am.
Yeah. Next participant, Mamta HC, UCS,
Yes, sir.
POSRS Technology, Tumkur University. Yeah. Please, can you share your slide?
Okay, sir. Okay, good afternoon, sir. Myself, Mamata HC, a research scholar from Tumkur University, Tumkur, under the guidance of Dr. Parimala B. Today, I am going to present physico-chemical characterization of Hanmantpura Lake, a seasonal study in Tumkur, India. Water is an essential component of an ecosystem and it sustains life on Earth. Roughly 1/3 of the Earth's surface is covered by water. About 97.4% of water is found in seas, nearly 2% of in frozen glaciers and ice caps, on 0.59% as a groundwater, and available fresh water is about only
Can you come to the result part?
0.01%.
Result part. What is your findings? You try to tell me your findings.
This is the my study area. This is the methodology.
Okay. We expect your findings only.
Okay, sir. This
Please come to that part.
result.
Yeah, okay.
Okay, sir. This is the water temperature and atmospheric temperature. The temperature is changes according to the seasonally. During winter season, there is a low temperature, both in atmospheric and water temperature. During the summer season, it will be high temperature, that is 28.45 about atmospheric temperature, and water temperature is about 27 degree centigrade. And during pre-monsoon and post-monsoon also. Then come to the electrical conductivity and turbidity. Here also, during summer season, that is it will be very high. That is electrical conductivity is about 1173.28, and very less in post monsoon 366.02. Then come to the turbidity. It can be moderate or it can be slightly high in during the summer season compared to all the seasons. Then come to the total dissolved solids, that is TDS. TDS also high in during summer season. Then is againly coming down during monsoon season, and it will be
Sorry
moderate
to
during the
you.
winter season.
Mamta,
Sir?
can you tell me your conclusion or summary?
Okay, sir.
Yeah.
This is total alkaline calcium
Just you skip.
Okay. The physical chemical parameters affect the quality of water and biotic flora and fauna. The seasonal values of water quality index indicate that during summer season, lake water is more affected than winter. The maximum values of temperature is 24 to 20 degrees centigrade, and pH is 8 to 9. Turbidity is about 280 to 400 NTU. EC is 1152, 1182. Then TDS is about 670 to 693 mg per liter. Total alkalinity is about 250 to 300 mg per liter. Chloride is about 150 to 174 mg per liter were observed during in summer season, while minimum values were observed in winter season. That is, temperature is about 21 to 20 degrees centigrade, and pH is 7.6 to 7.8. Then turbidity and as well as the electrical conductivity, total dissolved solids, total alkalinity, chloride, and about. And then moderate values are also observed during in the monsoon season. This study concludes that seasonal variation is driven by the rainfall and temperature, is significantly affect the physical chemical parameters of water. Summer
Okay,
season
ma'am. Thank you, Mamta.
Okay.
Actually, we have seen. I would like to ask you a very simple question.
Yes, sir.
What is the difference between weather and climate? Because you studied seasonal study, yeah. So that I'm
Yes, sir.
asking, what is the difference between weather and climate?
Climate is the our surrounding is called as climate, sir.
Okay, weather.
Weather overall means I think climate it is one of the conditional studies, sir.
Right. One more question, ma. What is the
Yes, sir.
difference between climate change and global warming?
Climate change enormously increase. Climate change it will be changes in climate. It will be increasing the global warming.
Okay. Thank you so much, Mamta. Thank you.
Okay, sir. Thank you, sir.
We'll move on to the next. Dr. S. Uttira Swami, Government Arts College, Salem. Are you here? Dr. S. Udira Swami? Okay, it's not available. We'll move on to the next one. Dr. M.I. Farhina, Government Arts College, Salem. Farhina, are you there? Okay, no response. We'll move on to the next participant. Suresh S. Sastra University, Tanjavur. Suresh, are you here? We'll move on to the next one. Vinita, Bishop
Yes, sir.
Heber College.
Yes, sir.
Yeah, please.
Yes, sir. Sir, is it available, sir?
No, my
Sir?
your screen is not. Share your screen, ma.
Yes, sir.
It's not visible.
Sir, it is now available.
No, ma, it's not visible. Not yet. Okay, proceed, ma.
Okay, sir.
Yes.
So my name is Vinita from Bishop Heber College. Sir, is it audible, sir?
Yes. Just brief your introduction part, then next move on to methodology and result, ma.
Yes, sir. My topic is about 9 anthracene carboxylate chief base synthesis, characterization, and its biological activity. And the introduction is based on chief base ligand 1 and 2 was I successively synthesis by using spectroscopic techniques like NMR, UV visible, and IR spectrum. In this structural conformations, I detect by IR spectrum. In this C double bond N, that is nothing but imine bonds. In the biological activity, both the ligands show biological activities, antibacterial and antioxidant assay. And the keywords I included are 9-anthracene carboxylate, characterization, and its biological activity. And the importance of the chief base is it is a type of a chemical compound and it is derived from various aldehyde and ketones. And the importance of the chief base is transition metal, which is derived from amino acid. And then I synthesized two ligand-based complexes. First one is 2-amino-4-chlorophenol and then methyl-2-amino-phenol. And then I used a chemical is 9-anthracene carboxylate, 2-aminophenol, 4-methyl-2-aminophenol, and copper chloride dihydrate. And then the method I used is synthesis of complexes by using amine plus aldehyde in the ratio of 1:1. In this, I refluxes with ethanol and glacial acetic acid. Addition to that copper chloride, the complex formation will appear. But in this complex formation, I recrystallized by using the ethanol. This is my complex. Methanol fused with the glacial acetic acid. I give this compound. In this compound, the N double bond C, nothing but imine compound. And then this is characterized by using, that means I confirm by using UV visible. In this, I obtain pi to pi star and N to pi star transition. Imine, I confirm this is the imine compound by the range of 1618 centimeter inverse and proton NMR at the range of delta value at 8.85 ppm. And this is a graphical representation for the spectrum. First one is the UV spectrum. I confirm this the L1 and the L2 decon. And the second one is the IR spectrum. And third one is the chief base for NMR spectrum. And then results and discussion. In this UV spectrum, there are in first ligand L1 show the absorption range in the following ranges. And then L2 ligand show the following ranges. I confirm that pi to pi star transition and N to pi star transition. I observe that it is an aromatic ring or conjugation is present. And then IR spectrum in this imine C double bond stretching at the range of 1618 centimeter inverse. I confirm that This is a shift base and the OH stretching at the range of 345. I confirm this is the OH stretching present in it, and then proton NMR in the range of 8.85. The delta value is 8.85. I confirm that is the imine proton. And then biological activity show ligand 1 and ligand 2. Both are antioxidant and antimicrobial effect. And the DTPH assay also is done. In this, antioxidant activity rises from 44 to 47%, and IC50 values also done. It first ligand 42.28 μg per ml it is obtained. And then conclusion is shift-based ligand L1 and L2 was successively determined by using NMR, UV visible and IR spectroscopy. In the both ligands show biological activities like antibacterial and antioxidant test. L2 ligand is better compared to the L1 ligand. Any questions, sir? Sir?
Yeah, yes, I'm here. Your work in what way it will be very useful for the scientific society?
Sir, in chemistry point of view, sir,
Okay.
I can synthesis the complex, sir.
Okay.
And I confirm this is the biological activities. I synthesis the compound. It has a both biological activities, sir. It is useful
Right.
for
Useful for
useful for people, sir.
okay, ma. Okay. Thank you.
Thank you,
Vinita.
sir.
Thank you. Yeah, next participant, Dr. Mohammed Ilanq Ahmed Khan from.
Yes, sir.
Yeah. Bhiwani, Haryana.
Yes, sir. I am ready, sir.
Yeah, your time starts now.
Okay, sir.
Kindly share your slides, sir.
Yes, sir. I am.
Madam.
Sir, it's visible, sir?
Yeah, yes, visible.
Yes, sir. It's very good afternoon. So
Very brief your introduction part and move on to methodology and result part, sir. Yes, sir.
Okay, sir. Thank you, sir. So very
Yeah.
good afternoon to all the members present here. So my topic is on role of nitric oxide in breast cancer. So I want to just correlate how this nitric oxide is going to play important role in the breast cancer. So before going to detail about all those things, just I want to inform you that breast cancer is an estimated 620,000 deaths globally around the world. And it is nearly half of the breast cancer occurs in the women also. As we know, it is the most common cancer in almost all the country. Out of the 185, it is occur in 157 countries. So it is a very drastic problems in whole world. And in men, it is a only 0.5 to 1% of the breast cancer.
Kindly move on to methodology and result part, sir.
Okay, sir, just it is because hypothesis, so I don't have this research. Just I want to tell the ideas. So what the nitric oxide do during the breast cancer?
So next slide, sir. Can you move on to the next slide?
Breast cancers and death is also very high in India because of the breast cancer. So when we talk about the role of nitric oxide, basically as it is a signaling molecule, as we know, because this play a very important role, and this nitric oxide have a dual role in case of the carcinogenesis, either acts as a tumoricidal as well as tumor promoting also. So I want to say you that there are three different isoforms of the nitric oxide synthase, which secretes nitric oxide or which produce nitric oxide. And the data shows that we have done those things also. These nitric oxide synthase are present in almost all the types of the cancers, like breast cancer, cervical, CNS, head, neck, gastric, and lungs. And we have also protected that the expression of the nitric oxide synthase is there in all the carcinogenesis. When this nitric oxide and reactive nitrogen species reacts, they cause DNA damage, and because of this cell signaling, it also causes the DNA break, leads to the GC280 mutations, and leads to the 53 inactivation, and also suppress caspase activity. So these nitric oxides also do the angiogenesis by the promoting tumor growth, and this also acts as a VGA for the CGMP pathway. So this nitric oxide have a role in the different type of the cancer, even though in the breast cancer also, this is the pathway how they can regulate upregulation of the MMP, anti-apoptotic, angiogenesis, and
Sir,
down
excuse me,
regulation
sir.
of the.
Sir, kindly share
Yes,
your uniqueness of your work, sir.
okay, sir.
Yeah.
So I want to biotechnological innovations. Why I want to discuss this one? Basically, we can develop the nitric oxide donors and nitric oxide drug hydrides, okay, which acts as a chemotherapeutics and radiosensitizer. Basically, my idea is to present this one because of this. So, which
Sir,
will
1
help
minute.
you.
Sir,
Yeah.
whether
Yes,
whether
ma'am.
it's review article or a research article, sir.
Yeah, it's a review article. Hello.
Okay, come to the conclusion, sir.
Yes, ma'am. So conclusion, nitric oxide research in cancer offer a blueprint for the rapid biomedical innovations and developments. So we both think we can bridge the cancer biology pandemic surveillances also, so we can overcome these problems by developing the biotech innovations and developing the benchmark for the breast cancer by using the nitric oxide as a chip or nitric oxide as a some sensor-like instruments we can use. In that, we can put the nitric oxides molecules, so which will help in detecting the breast cancer. So this is actually idea behind the. This is our reference. So thank you, ma'am. Any question?
Sir, that was a presentation which actually creates an alarm for us. And do you have any hypothetical methodology for that, for this work to incorporate it in future, sir?
Yes, ma'am, because there are some these nitroxide synthase enzymes are there which express in all the types of the cancers. So we are actually doing something so to see which actually NOS is involved in the breast cancer basically, so that we can produce some benchmark for that, and we can go for some development of the sensors which helps to detect the early breast cancer. This is actually
Okay. So in that case, in future, while presenting, that's a small suggestion from our side. Like you can just give a small layout of what you're going to extract from your review. So that would be quite useful for us. That was a very good presentation. Thank you so much, sir. Thank you.
Okay. Thank you, ma'am.
Good evening, madam.
Next participant, Punita, are you ready?
Ma'am, Muthiraswamy is here, madam.
Yes, ma'am. Yes, ma'am.
Ma'am, Muthiraswamy. Ma'am. Ma'am, Muthiraswamy is here, ma'am.
Kindly wait, sir. Punita, are you ready?
Yes, ma'am. Shall I share my screen, ma'am?
Yes, ma'am.
Ma'am, it's showing like host disabled attendee screen sharing, ma'am.
Now you can share.
Okay. Better visible, ma'am?
No, it's not visible, ma'am.
Whether it's visible, ma'am?
Yes, yes.
Okay. Good afternoon, one all. Myself.
Kindly move to materials method and then your result discussion part.
Okay, ma'am. Just a small brief. Foraminifera or tiny unicellular protists that can be found in all type of marine environments. They can be used as a biomonitoring tool. My research objective is to access the diversity of benthic foraminifera and to determine the live and the dead population of benthic foraminifera and to evaluate the sensitivity to metal concentrations. This is the study area which involves three islands. First one is Upper Island, next second one is Walimune Island, and the third one is Anaipar Island. The methodology involves a collection of water and sediment samples. The water samples has been tested for temperature, pH, dissolved oxygen, salinity, TDS, conductivity, and turbidity in the field itself. And the sediment samples has been tested for sand, slit, and clay ratios and calcium carbonate and organic matter contents. In the field itself, 100 grams of sediment samples has been stained with the rose Bengal in order to determine the live and the dead populations during the sample collection. After a period of 1 month, these samples has been washed to remove the excess stain. After then it is dried and the foraminiferans are picked with the help of a stereo microscope, and it is mounted and it is a photography has been done. The heavy metal analysis has been done by atomic absorption spectroscopy. This is the methodology I have followed. This is the sampling procedure where the samples were collected in the field itself and had been stained with the rose Bengal. The results shows that the foraminiferal, there are the Ammonia baccari and Elphidium crispum and Quinquilona semilunum are the major species which has been found in all the three islands. We have collected two samples from each islands, and the live and the dead populations were calculated. First island shows the highest population when compared to the other two islands, and the dead species were found in more in the third island. The pink color, the image shows the pink color, which is found to be alive at the time of sample collection, and the at the down, which is not stained with pink, is found to be the dead organism, which is found at the time of sample collection. These are the various water parameters and the sediment parameters. The water parameters such as water temperatures showed a positive correlation with the foraminiferal eminence, and the salinity and the pH showed a moderate positive correlation with the
Mika, 1 minute.
foraminiferal.
What is the economic importance of this species, ma?
This is used as a biomonitoring tool, ma'am. If the species is found in more numbers, the island is found to be in a good condition. Whether the population is found to be less, there would be some pollution or some other anthropogenic activities. This can be identified with the deformities which can be found in the structure of a species, ma'am.
Your conclusions, ma?
Conclusion is that foraminifera communities will respond to the changes. A very little change can be absorbed by this organism. Will it give rise to a nutrient tolerant species as well as of the cold tolerant as well as the metal resistant species. This will help as a biomonitoring tool rather than using of physical or any other chemical methods. We can use this organism as an excellent biomonitoring tool which does not cause any harmful to the environment.
Okay, so where did you do your collections, ma? Your species
Yes.
collection?
It is from Gulf of Mannar, ma'am. In the Gulf of Mannar, I have chosen three islands, ma'am. I have also got the forest permission because it is a marine biosphere reserve. I have also got the permission in order to collect the samples, ma'am.
Okay, so you yourself went directly and you collected.
Yes, ma'am. Myself and my guide, along with the scuba divers, we went, and along with the forest officers also, ma'am. We went and collected the samples, ma'am.
First of all, congratulations for your effort, for the passion which you have in doing this particular experiment. Being a
So,
girl, you have traveled so far and you have done, and congratulations from our side for this
thank you.
beautiful work. So my
Thank you.
question is that, like, um, did you authenticate the result as a bio indicator? Is there any evidence for that?
Yes, ma'am. It's a bio indicator. There are many papers which says it as a bio indicator.
Okay, so from your side, do you have a futuristic perspectives for that?
Yes, ma'am. We have to prove it by doing various any other type of study, we have to. I'm just at my preliminary stage of my work, ma'am. So I have to work on it.
Okay. So all the best, Punita. That's a great work.
Thank you.
Congratulations and appreciation to your guide also.
So
Thank you.
thank you so much, ma'am.
Madam,
Next
Uttaraswamy is
is
here, madam.
Vishnupriya.
Madam,
Vishnupriya
Uttaraswamy is here,
Krishna.
madam.
Vishnupriya from Sri Krishna Arts and Science College. Vishnupriya from Sri Krishna Arts and Science College.
Madam, 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 are you get
Vishnupriya,
not
are
here,
you
madam?
there? Sir, please wait, sir. Vishnupriya, are you there? Okay. So Pradeep R, Bishop Heber College.
Hello, ma'am. Yes, ma'am.
Pradeep, can you please
Yes, ma'am. Okay, ma'am.
share your slide, ma'am?
Ma'am, are you visible?
Yes, Pradeep, you can proceed.
Thank you, ma'am. Ma'am, I am Pradeep from second MSc Chemistry. I'm going to present my work on exploring the role of para-substituted in fluvonides binding in 6LU7 via molecular docking, ma'am. Fluvonides are well-known class for natural polyphenolic compound. They are widely studied for their antioxidant, anti-inflammatory, and anti-cancer activities. The protein we used is 6L U7. It play a critical role in viral replication, and it also making it prime therapeutic target. Materials and methods. For this study, I use autodock vena for molecular docking, and also I take protein from protein data bank, and I also use Chemtra Ultra 12.0 and Chem 3D Pro 12.0 for minimizing the structure, and also I used the autodock tools, and also used Discovery Studio for binding. Methodology is first
Do you have any
preparing the
workflow for
protein by removing water molecules and adding the hydrogen, and then prepare the ligand with the what, ma'am? Yes, ma'am.
Do you have a
Ma'am?
workflow for your methodology?
Workflow?
Pradeep, do you
No,
have a
ma'am.
workflow for your methodology?
No, ma'am. No, ma'am.
Then you can go to your results, ma. We understand this.
The fluorosubstituted flavonoid showed the strongest binding affinity, ma'am, with the docking score of 7.4 kilocalories per mole, ma'am. And the chloro and bromo substituted flavonoids show the good affinities around minus 7.3 kilocalories, ma'am. And these are the complex molecular surface 3D and 2D structure, ma'am. And the conclusion is the fluorosubstituted flavonoid was identified as the most promising candidate, ma'am. It is it is used against the antiviral drug against as a COVID-19. Thank you, ma'am.
Pradeep, thank you. In future, when you are going to present a docking studies or something, put a formalized workflow first. That's very important. And then tabulate your results. Tabulation is very important in docking.
Ma'am,
Apart
are you, thank
yeah,
you.
apart from minimal score, there are so much of other parameters
ma'am.
in docking. So please concentrate on that. Thank you.
Okay.
Ma'am, excuse me, ma'am. I have a meeting, madam. Please, you understand my situation, ma'am. Madam, ma'am, excuse me,
Hello?
ma'am.
Yes, yes, yes.
Sir, I am
Uttaraswamy is here.
Uttaraswamy here. Yes, sir, Uttaraswamy, sir.
Yeah, yes. Are you ready, sir, now?
I am.
Yeah, please go ahead. Go ahead. Please share your slide. Sir, please cut short your presentation. Hello, sir. Dr. Uddiraswamy, sir, are you here? Sir, your screen is not visible, sir. Hello? Uddiraswamy, sir. Monica. Next, we'll move on to another participant, Dr. Monica Jangra, Chaudhary University. Dr. Monica.
Yes, sir. I am audible.
Yeah, please, your time starts now. Please share your screen.
Sir, excuse, sir. Sir.
Sir, yes, sir, yes, sir. Sir, actually, another ma'am is sharing her screen. Kindly wait, sir. After this presentation, you can proceed, sir.
Is my screen is visible?
Yeah, yes, yes. Please, you can share your slide. Your slide is not visible, madam. Your screen is not visible. Please share your screen. Uddiraswamy, sir, kindly wait for 5 minutes. After this presentation, you can go ahead. Monica,
Yes, sir. I am trying to share my screen.
please share your slide.
Is it visible?
No, Dr. Monica, it's not visible, not yet. Dr. Monica?
Now is it visible?
No, not yet visible. We are waiting for your
Sir, it is not sharing.
Dr. Monica, if you are unable to screen,
Yes, I
kindly
am
wait.
unable to share the screen.
Now the session will continue after lunch.
So
Okay.
let it run, sir. Let it try now, sir.
Yes, please. Udrasami, please, you go ahead. Please share your slide. Dear participants, is there any technical issues? We'll continue after the lunch.
Sir.
Yes, please.
Sir, not working, sir.
Okay, sir. Don't worry. You go and have your lunch and come back.
Hello, sir, may I interrupt you?
Yeah, Seema Rani is here.
Yes, can I present my work as I have an important meeting at
Kindly,
2:30?
yeah, kindly proceed, ma'am. You
Okay,
can share.
please
Yes.
freeze Heber so that I can share the screen. Okay.
Dr. Seema Rani, kindly share your slide.
Yes, I am presenting.
Yeah.
Is my screen visible?
Yeah, yes, visible. Please, you can
Okay.
proceed, ma'am. Very brief
Hello
yes,
everyone.
presentation,
The
ma'am.
topic of my presentation is unraveling the role of adhesion molecules in vitiligo pathogenesis. As we know, the skin is mainly consists of two layers, that is epidermis and dermis, and epidermis is mainly formed of melanocytes and keratinocytes, whereas dermis is formed of fibroblasts. The skin pigmentation is regulated by the melanin pigment, and this melanin pigment is responsible for the uniform coloration of the skin. If there is disturbance in the melanin pigment, then there is white patches occur in the skin, and these white patches are called the vitiligo. Vitiligo is characterized by basically the loss or disappearance of melanocytes from the skin. This skin pigmentation, in this skin pigmentation, we require that is dermis and epidermis for the uniform coloration of the skin.
Hello,
In
madam.
present study, we
Kindly
have
share your.
performed, we
Hello.
have concentrated on adhesion molecules. These are the cell adhesion molecules. These are basically responsible for the cell and cell extracellular matrix binding. These adhesion molecules are integrins, immunoglobulins, selectin, and cadherin. Basically, these important for the cell growth, cell differentiation, cell organization, and cell and cell to cell interactions. In the present study, we have taken 15 vitiligo patients and 8
Madam,
control.
your screen is not moving. We can only say one word, graphical
Okay.
representation.
Okay.
Your screen is actually freezed.
Madam, no issue. Can you share your uniqueness of your work? Madam, your presentation is not needed. Kindly share your work, madam. Highlight your work. It's enough.
Actually, we have taken two types of patient, vitiligo patient and the two types of vitiligo patient, that is active vitiligo patient, stable vitiligo patient. In the active, Hanji?
Hello,
In the
madam.
active vitiligo patients, we have worked on the their.
Madam, kindly
Yes, sir.
highlight your work.
Is my presentation visible to you?
No, madam, it is not visible, madam.
Okay, sir. Actually,
Madam,
I am
it is not
highlighting.
needed, madam. You can brief your work, madam. It is enough. Come to the conclusion
Okay.
part, madam. What you have done.
Okay, sir. I
Yeah.
have actually performed the real-time PCR and protein expression of ETS, that is transcription factor, E-cadherin, ICAM, and integrin alpha 1. I have analyzed that ETS expression was found to be very low in the vitiligo patient, and the expression of extracellular matrix and adhesion molecules are also very found to be very decreased. Then I have analyzed, that is analyze scanning electron microscopy and the adhesion assay. Scanning electron microscopy showed that melanocytes have no dendrites and very large perinuclear space, whereas adhesion assay say that there is lacking in the adhesion of cells with the substratum of the melanocytes. Then we analyze the extracellular matrix component, that is collagen, type 4, elastin, and fibronectin, and we found their expression was found to be significantly decreased in the lesional skin of vitiligo patient as compared to control, as compared to stable vitiligo patients. In the conclusion, we can say that the decreased zero expression of ETS1, that is transcription factor, and low expression of adhesion molecule may be responsible for the loss of melanocytes and failure of repigmentation therapy in the vitiligo patients. Thank you. Is there any questions regarding the?
Thank you, ma'am.
Okay, thank you.
Madam, if I will try, madam. Madam.
Yes, sir.
Uttarasamy sir, are you able to share your screen? Who is ready to share the screen?
Now I'm. Can I try to share my
screen? Monica?
Yes. Yes, ma'am.
Monica, proceed, ma. Kindly go to materials method and then result and discussion.
Now it is visible?
Yes.
Yes. Good afternoon, ma'am. Thank you so much for giving so much time. Today my topic is the weather-driven changes in the chili mite, Polyphygotarsonimus latus, Acari tarsonimedi. That is a study of the chili mite in chili agriculture. So when we're talking about the chili mite, it is a very small mite. We cannot see in with the visible naked eye. It feed on the sap, and that is present under side
Monica,
of the
your
leaves.
screen is not moving, ma.
Is it visible?
Okay, okay.
When we're talking about their adult mite, it is also known as the broad mite, yellow mite, chili mite, due to its yellow color or amber in color. They have the 8 pair of legs, 4 pair of legs, swollen bodies, light yellow color. Male have the smaller as compared to the female. In the case of the male, they have the enlarged hind legs that used to pick up the female nymph and place her at right angle to the male's body for their later mating. If we're talking about, there has a very drastic symptoms when they infest the chili crop. Stunted growth may be seen, elongation of petioles of the older leaves, and inverted and boat shape of leaves have the appear during the heavy infestation of the P. latus. That is also known as the Mudra complex disease. So, according to our methodology, we have done this study in the research farm area, Department of Vegetable Science, Chaudhry Charan Singh Haryana Agriculture University, Hisar, and replication were taken in the 3 replicates. Plot size is the 3 into 2.7 meter, and spacing in between the plants, that is the 60 centimeter into the 45 centimeter. Firstly, we taking the leaf from the top, middle, and bottom from the 10 randomly plants, and taken these leaves to the acarology lab, and where we study under the stereozoom binocular microscope, and their observation will continue up to the till harvesting of the crop. After that, the meteorological data was taken from the Agriculture University, Agriculture Meteorological, CCS Haryana Agriculture University Department, and the maximum and minimum temperature, morning and evening relative humidity, rainfall, sunshine hour, evaporation, wind speed, all this data have the observed during this study, and the correlation matrix was find out in between the abiotic factor, and P-latest incidence. Then overall the result we have concluded that in the month of the October, the maximum number of the mites were observed during the both time period in 2015 and 2016. That is the due to the high maximum because it favor the temperature in between the 30 to 34 degrees Celsius, and it may be minimum temperature is the 18.9 degrees Celsius. Relative humidity was the 82%, and the evening relative humidity was the 29.4%. Sunshine hour was there 8 hours. Evaporation was the 4.4 mm, and similar result was observed during the 2016. So we can say that the maximum temperature is the significant as compared to the this, and the minimum temperature is non-significant. Relative morning relative humidity is significant but not beneficial. So at last we can conclude that there is a very hard effect of the this study include that there is a meteorological parameter of the population
Rika,
of the
your
yellow
time's up.
Yes, ma'am.
Tell your conclusion.
Yes, I am concluding that the study conducted to determine the influence of the meteorological parameters on population of yellow mite P-latest on chili revealed that the mite population have positively correlated with the maximum temperature and negatively correlated with the relative humidity. Minimum temperature, wind speed, sunshine hour had no significant effect on the population dynamics of the chili mite. So we can use this developed forecast model for the P-latest infestation. Thank you, ma'am. If you have any questions.
Okay, ma. Thank you. Thank you. Uttarasamy sir, are you here? Anybody else want to share your screen? Otherwise, we will close the session. Hariharan.
Yes, ma'am. Can I share, ma'am?
Hariharan, your next session, ma. You are in the next slot.
Ma'am, Suresh, classes, ma'am.
13, ma'am.
Okay, thank you. Thank you, participants. We have a technical talk now. We will continue the next oral presentation in the afternoon session. Thank you.
Ma'am. Suresh, ma'am.
Madam.
Students, please occupy the front row. Students, please occupy the front row. We are about to start the keynote session 2. Students, please occupy the front seats. Students at the back, take your seats. Students at the back, please take your seats. Students, please occupy the front row. Students, please maintain the decorum and occupy the front seats.
Okay. You can start.
என்ன சொல்ல
Students, please settle down fast.
Students, please occupy the front row as the keynote session 2 is about to start. We now commence keynote session 2, which will be chaired by Dr. S. Sriram, Assistant Professor, Department of Biotechnology. Please, sir.
Good afternoon, students. Students, good afternoon. It's time to settle down. Camera on. Only photos. Video is going on. The photo. Video. Camera. Live போயிட்டு இருக்கா? யார் அப்ப நடுவில் நிக்க சொல்லுவாங்களா?
Students, kind attention. Yes, we are about to start the next session. I request you to all settle down in your respected seats as we are about to begin the keynote session 2.
So good afternoon, students. Welcome back to the afternoon session, technical session 2 for this national conference sponsored by Government of India. So it's my pleasure to introduce the speaker, Dr. Edwinder Singh, Assistant Professor, Department of Botany, Central University of Punjab, Bathinda. So Sir has great experience in different fields. He has started his BSc in Medical, followed by MSc Botany, PhD in Life Science, specialization in Cyanobacteria, extremophiles. So we are talking about extremophiles. Sir has worked in cyanobacteria. He has screened for different cyanobacteria species from hot springs as well as in high altitudes. And his main focus is working on prokaryotic microalgae. And he has published 25 research articles. And most of the articles are there in a core subject, Journal of Applied Phycology and Algal Research. They are the topmost journals, especially in this field, phycology field. Later on, he has got funding from DST SERP of about 43 lakhs. Again, it is in a microalgae. SAR has over 1,000 sequence. He has published more than 1,000 sequences of extremophiles of cyanobacteria in NCBI database. Now, when I was discussing with SAR, he said he has around 500 strains of extremophiles, which is an untapped resource. It is apt for our topic, Prosperous of India. This is the best resource for secondary metabolites. And we are fortunate to have our SAR to present this topic on algae nanoparticle production from algae. Thank you, sir, for visiting our institute. Hello.
So, good afternoon, all. Dear students and my dear colleagues sitting in front of me. Good afternoon. And first of all, I want to thanks Department of Biotechnology, Bishop Heber College, TIRUCHIRAPPALLI, especially head of the department, Ma'am Jasmine, Dr. Danish, and Ma'am Priyadarshini, to invite me. And they contacted me last year, and he are directly in contact with me, so that it will be easy for me to join here. So it is my privilege to be here. Although it's my first visit in this college, but I have visited Trichy 10 to 12 times before visiting there. So as sir told, I am started my career as a extremophile taxonomist. In my PhD, I have worked on hot water springs and cold deserts regions of northwestern Himalayas. And after completing my PhD, in my first job that I worked for 7 to 8 years in one university, where we have worked basically on mesophytes that are wetlands of Punjab, basically on a microbial ecology aspects. And then in between, one student came to me for PhD. He is more interested in nanoparticles, although it was not my core area. I am algal biotechnologist. So basically, we are working on a biopolymers from algae, and as a source of bioplastics, and for the you can say biopolymers to protect you can say as a skincare products. So this is one of my area where we are working on. So this is work where I going to present here. It is work of my one PhD student who has recently submitted his work. So first of all, I want to acknowledge him for his hardship he has done for his PhD for last 4 to 5 years. So now coming on topic, I have chosen topic according to the theme of the conference, that is you can say future pandemic preparation and emergency response through biotechnological innovations for a prosperous India. So for this main theme, I have chosen the sub-theme of my presentation, that is algae as a sustainable source for nanoparticle synthesis with novel application. In this presentation, we will discuss briefly about the nanotechnology, and then on the why we have chosen this algae as a source of nanoparticle synthesis, and one part of his PhD work for a copper nanoparticle synthesis using one macroalga, that is a cladophora. So not taking much time, we started with, you all I think well aware with the term nanotechnology. You know that what is nanotechnology basically is a nanoscience, is an application-oriented research comprised different field of physics, chemistry, and biology. So in which we deal with the particle size from 1 to 100 nanometers. So first, Richard Feynman was the first physicist who explored the prospects of manipulation of atoms and molecules in his lecture on the plenty of room at the bottom. So whereas Nora Taniguchi is 1974 first used the term nanotechnology, to engineer the material at a nanoscale. So this is the brief, you can say, introduction about the nanomaterials. So nanomaterial are nanometer size particles that are nanoscale in 3 dimensions. They include nanopores, nanotubes, quantum dots, nanoshells, nanorods, nanospheres, nanowires, nanorings, and nanocapsules. So nanoparticles are not justly simple one molecules, but rather a combination of 3 layers. So these are surface layer, shell layers, and core layers. So shell layer is which is maybe a functional with a variety of small molecules, metal ions, surfactants, and polymers, whereas the shell layer, which is a chemically different from the core in all aspects, whereas the core layer, which is essential as the central portion of the nanomaterials. So in recent time, nanoparticles have gained too much attention from the biologists as well as the material biologists. So because it is having a wide range of application due to the matter at this scale present a more compact arrangement of atoms and molecules that generating phenomena and requiring enhancing mechanical, electrical, magnetic, optical, catalytic, and antibacterial properties that are completely different from their counterparts, that are the macromolecules. So by reducing the size, nanoparticles have a higher surface to volume ratio, enabling a greater number of atoms or molecules per volume, which means that less material is needed to obtain a same activity and exhibit other properties. So they can classify it based on their composition, shapes, and size. The most common type of nanoparticles are metals, metal oxide, carbon-based, and quantum dots. So owing their unique size and properties, they have attracted significant attention in various fields, including medicine, electronics, energy, and environmental science. So this picture categorizes the nanomaterials. These are 0 dimensions, 1 dimensions, 2 dimensions, and 3 dimensions. 1 dimension include nanoparticles and fluorescence, whereas 0 dimensions and 1 dimensions include nanotubes, nanohorns, whereas 2 dimension nanosheets, nanolayers, and 3 dimension layer is nanowire array and nanotubes. So according to approach of synthesis, we are using basically 2 approach for synthesis of nanoparticles. One is bottom up approach and second is top down approach. So bottom up approach is a chemical method or a biological methods are applied. So chemical methods are including sol gel, spinning, and chemical vapor vaporization, whereas in biological method, as named, different biological sources are used in vitro as well as in vivo synthesis of nanoparticles. Whereas the top-down approach, physical and chemical method is a laser ablations and 3-dimension decomposition. In this presentation, we will mostly talk about on the biological method for synthesis of nanoparticles. These both approach, bottom approach and top-down approach, are differentiated on the basis of in bottom approach, the nanoparticle built from a small entities, for example, by joining atom molecules and small particles. So in this approach, the nanostructure building blocks of the nanoparticles are formed first and then assembled to produce a final particles. So this approach mostly lies on a chemical and biological synthesis, whereas top-down approach, nanoparticle produced by size reduction from a starting materials. So size reduction is achieved by various physical and chemical treatments. This method induces imperfections in the surface structure of the products, and this is the major limitation because the surface chemistry and other physical properties of nanoparticle depends on the surface structures. So different type of processes are categorized in chemical methods. You know that using a chemical reactions. So this second one is the coating process, and then finally synthesis. Whereas physical method, laser ablations used, and by using metal solvent reactions, different type of nanomaterials are produced. Whereas in biological sources, as a name can depict that biological entities as well as biological extract obtained from biological materials are used for the reduction of, you can say, for the production of nanomaterials. So different techniques after production, different techniques are used for the characterization, in which for the morphological characterization, SEM and TEM are mostly applied, and for the particle size, DLS, and then optical characterization has been done by UV and photoluminescence. That is the preliminary characterization technique that is employed to detect the reduction of nanoparticles. And then structural characterization is achieved by XRD, EDX, XPS, FTR, Raman, and surface area characterization by BET. and thermal property and stability are determined by the TGA and DSA. So mostly these techniques are used for the physical characterization of the particles produced. So we are mainly focused on the green synthesis. Why? Because the chemical synthesis that is not an environmental friendly approach, because synthesis of lot of byproducts that are not chemical, you can say environmental friendly. So biological methods are considered are more eco-friendly technique for synthesis of a nanoparticle, because it is a, you can say, you know that the biological, when we use a biological method, there is a less harm to the environment. So it is gaining a great interest in recent time, last 10 to 15 years. So green synthesis methodology basically based on the biological precursor, depends upon various reaction parameters such as solvent, temperature, pressure, and pH conditions. In biosynthesis, metal oxide nanoparticle based on the availability of effective phytochemicals in various extract of plants, microorganisms, including fungi and algae, especially in leaves such as ketones in plants, in ketones, aldehydes, flavonoids, amides, terpenoids, carboxylic acid, phenols, and ascorbic acid are present that act as a reducing and capping agent for the synthesis of nanoparticles. So these are few advantages for production of nanoparticles through biological approach, because they are biocompatible, one-pot method, easy to handle, simple and rapid production cost, cost efficient, and mainly eco-friendly. So this is the one flow chart that is followed for the synthesis of a nanoparticle, in which initially extract was made, or in which then metal mixed with the metal precursor, and then bioreduction by giving a specific temperature or a reaction conditions. So different type of nanoparticles synthesized, and then on the basis of characterization, its shape, size, and type has been depicted. So different type of applications, as we have known that it's getting attention day by day. Because of you can say it's eco-friendly natures. So mainly in the bioremediations, now and nanoparticles are not, we can say bioremediation, we can say the remediation of pollutant from the environment. So because it's having a high antioxidant activities, having a high antimicrobial properties, application in cosmetic and medicinal equipments, useful in targeted drug delivery, labeling nucleotides and peptides, and it also exhibit a great anti-cancer activities as well as the anti-inflammatory activities. Why we have chosen algae? As you are well aware with that, algae is a group of photosynthetic prokaryote and eukaryotic organisms. So that are photoautotrophs that can we found in a cosmopolitan in nature, in everywhere on this you can say on a earth. So we then know that algae have a significantly greater rate of tapping carbon dioxide through carbon dioxide sequestrations and hyperaccumulation of toxic metals, lack of hazardous bioproducts, low energy inputs, and richest supplier of various important biomolecules and nutrients. So the ability of algae to accumulate metal and reduce metal ion make them superior counterpart for biosynthesis. So the term basically as a new term emerged for the synthesis of nanoparticles using algae, that is FICO nanobiotechnology. So in recent years, so basically algae have different biomolecules, like another plants that are helpful for the synthesis of a nanoparticle using their counterparts. Different type of algal groups, algal organism belonging to different families has been exploited till date. So basically the approach, this is the some layout how algae is able to synthesize and what type of biomolecules are responsible for synthesis of nanoparticles using algae as a precursor. So this is taken from literature. So this is the one possible mechanism that are used by algae for the synthesis of a nanoparticle in vivo. This is proposed by Debajit, in which intracellular as well as extracellular when we produce a in vivo. The similar method, you can say molecules are responsible for reduction in vitro also. So intracellular method involve transporting ion into the microbial cells to the form nanoparticle in presence of enzymes. So in which fertile metal ions are absorbed by the cell membrane, then taken up by the molecules that are present on the surface of cell membrane, that by oxo-reductive reaction reduce the size of that metal ions. And similarly in extracellular, metal nanoparticles are in which proteins or different carbohydrates products are excreted outside the membrane that act as a precursor for the reduction of metal ions. So this is the after brief introduction. What actually now we are talking about the main, you can say, work that are carried on by Mr. Vinay, that is my PhD students, in which we have used a Cladophora glomerata, that is a macroalgae, that is a freshwater microalgae. We have this is organism that is available in abundantly in our, you can say, agriculture system in a rice fields. So we have collected samples from a rice field ecosystem and then and identified that organism as a Cladophora, and then used and make the extract from this organism and use this as a precursor for synthesis of a copper nanoparticle to determine their different biomedical application, including antibacterial, antioxidant, and anti-gas properties. So why we have chosen copper as a metal for the synthesis of nanoparticles? So copper has a lot of intention in the field of nanostructural materials because copper exhibits a confluence of advantage characteristics, including extensive surface area, precisely controllable redox behavior, and remarkable electrochemical performance alongside exceptional thermal conductivity and superior stability. So scientists have developed various methods to create copper oxide nanoparticles, include chemical vapor depositions, whereas gas phase react to a solid on a surface, whereas a liquid phase synthesis using solutions, the sol gel method using liquid that gel and then solidifies, and the colloidal synthesis using a suspension, and electrode deposition using electricity, and green synthesis by using extract of a plant, bacteria, fungi, and algae. So due to their affordability, because it's a less costly, natural prevalence, and microbial toxicity, copper emerged as a promising material for developing cost-effective and environmental friendly solution in both environmental remediation and pharmaceutical fields. So copper's unique properties such as stability of deactivate pollutant or act as an antimicrobial agent can be harnessed to design innovative technology for cleaning up contaminate sites and combating diseases. So research efforts are actively exploring a strategy to leverage copper's inherent advantage for a more sustainable and healthy futures. So one key property of copper nanoparticle is their p-type semiconducting behavior, which make them valuable for electronic applications also. So copper nanoparticles are demonstrate impressive catalytic biological activity, opening the door for their use in chemical reaction and biomedical applications. So the field of synthesized copper nanoparticles using agriculture is popular recently, driven by the promise of bio-inspired metal in multiple applications. So organism that we have chosen, that is a Cladophora glomerata. So this test organism is underutilized filamentous green macroalga with rectangular thalli, and readily available in both marine as well as the freshwater ecosystem with which highlights its ecological versatility. So according to previous study, Cladophora glomerata species is rich in biotic elements like phenol, fatty acids, strol, terpenoids, and carbohydrates. And these molecules have a potential to enhance the performance of copper nanoparticle synthesis in area like antibacterial activity, antioxidant properties, and even anti-cancer applications. So its potential as a source of cosmetic ingredient and antivirant for their future investigations. So furthermore, exploring glomerata open avenues for future research beyond the synthesis of a copper nanoparticles. To the best of our information, this was our first work to use a Cladophora for the synthesis of copper nanoparticles. So in which first we have collected samples from different habitats, including you can say wastewaters as well as a freshwater like agriculture field like a paddy fields. So then we have collected the you can say washed the obtained biomass properly with distilled water and identified the organism on the basis of their morphology as a Cladophora glomerata foods. And the collected algal species was then dried in a room temperature. So and then the extract was prepared from a dried you can say algae by pulverizing it, and pulverized algal biomass was mixed with the known volume of distilled water and placing on a 60 to 70 degree centigrade temperature with a continuous stirring for 1 to 2 hours, and the resulting mixture was then centrifuged and to get the cell-free extracts. And this extract was preserved in a 4 degree centigrade kept at 4 degree centigrade for their future applications. The phytochemicals, as we have told that earlier, that phytochemicals play a major role for the reduction of a metal to metal nanoparticles. So first we have analyzed what type of biomolecules are present in a prepared algal extract. For that, GCMS approach was utilized, in which we have found that although there are a number of peaks were obtained, but the 5 major peaks we have that belong to the oleic acids in lipid, tetradoinic acid of fatty acids, dodenosyl acetyls of ester, and then eosines as a saturated aliphatic hydrocarbons. That depict maybe these organisms play, these biomolecules play the important role for reduction of a copper to a copper nanoparticles. And for biosynthesis, the known volume of you can say algal extract was taken, and that were placed on a 0.01 molar of copper nitrate solutions at a pH 7 to 8 and 80 degrees centigrades. And after color change, it picked the reduction of from bluish green to grayish green solutions that show the reduction of a copper to copper nanoparticles. And that was the reduction was determined by applying a UV visible spectroscopy analysis. So this is the primary characterization techniques to depict the reductions. So the collided suspension of finished products displayed a substantial absorbance at 264 nanometer, which served as a, according to literature as well, the proof for the you can say fabrication of a copper nanoparticles. So the absorption band at 264 nanometer was thought to be caused by the interband transition of copper metal core electron observed in a copper oxide nanoparticles. So the conversion of copper metal to generate a copper nanoparticle driven by the occurrence of components like protein in the cell-free extract of the test organisms. Furthermore, the type plot data was utilized, which depict that the band to identify the band gap of energy of copper that is found to be 3.7 electron volt. And further, the synthesized product was characterized for their structural properties by using FTR methods. So surface functional groups of green produced nanoparticle explored. So the green fabricated copper nanoparticle spectra exhibit significant fabrication at 538, 588, and 1113, which according to literature as well as our previous study showed the synthesis of a copper oxide nanoparticles, whereas peaks at other surface show that their attachment of biomolecules to the metal ions. So that are belonging to the OH stretching as well as the COO stretching. So on the other hand, the peak at correspond to polysaccharide proteins, lipids, as well the peptidine chain respectively to the Chlamydophora glutamata extracts. So we also performed a FTR analysis of a extract that are obtained from a Chlamydophora that showed that matching by the peak, it showed that maybe the biomolecules that are present in the extract played a major role for reduction of a copper nanoparticles. Further XRD analysis depicts by using a JPS SD card 969016325. So the synthesized material was observed that is well crystalline and free from any impurities, as evidenced by the absence of any further phase and sharp intense peaks. Those standard deviations approximation was utilized to estimate the typical crystalline size of the nanoparticles that was determined to 18.1 nanometer. Further, the structural characterization was done by using a FESAM, HRTEM, SAD pattern, and EDS spectra. By using FESAM and HRTEM, the structural morphology of copper nanos were examined. It is evidently observed that the morphology of biogenic synthesized copper oxide nanoparticles is sorted spherical, with particle size between 40 to 50 nanometer. The SAD pattern displayed a sharp diffraction region, demonstrating their biogenic fabricated nanoparticle composed of single crystal, which are reliable with the XRD results. So weight composition of copper that is 81.59%, and oxygen 18.59%, and the atomic composition of copper 52.44 and oxygen 47 was measured by the EDS. That also show that the formed material was a oxide of a copper. The BET study was utilized to study the surface area, pore volume, and pore size of the green synthesized nanoparticles were determined to be 34.22 meters square gram per gram and 0.011 cubes centimeter per gram and 3.3 nanometer respectively by BGS assessments. The finding revealed that the mesoporous character of the has produced copper oxide nanoparticles, whereas XPS analysis of copper 2p area display peak the bending energy between 932.1 volt and 952.2 volt, which has been assigned to be a copper 2p over 2 and copper 2p1 over 2. So in OS1 oxygen and 1s core level spectra, there are two peaks visible in the fitting of curve at 529.2 and 530.4, whereas the crystal structure is responsible for peak at 529. 529.2 volts. The peak at 530.4, however, result from the chemo-absorbed oxygen attached to the surface adsorbed hydroxyl molecules. So fitted curve to the C1s region, it has a peak at 284.3 to 85.2 volt correspondingly, which caused by the presence of CC and carboxylic bond. So TJ thermogravitic analysis was utilized to determine the amount of biomolecules that are attached to the surface of the nanoparticle, as well as the, as you can say, thermal stability of the prepared materials. So initial weight loss of almost 3.9 happened between room temperature and 200 degrees centigrade, which coincide with the water absorbed on the surface of the copper nanoparticles evaporating. Another weight loss of nearly 4.8% was noticed from 200 to 8 degrees centigrade temperature associated with the organic matters degradation of a copper oxide nanoparticles that show that exactly the biomolecules that are performed as a core shell of that nanoparticle synthesized using a cladophora biomass. Further Raman spectra, the Raman spectra 3-dimensional 1-photon mode were detected. 3 peaks correspond to vibration mode of AG 293 centimeter and BGF 340 and 621 centimeters. The wavelength found in this investigation was nearly identical to the value of monoclinic copper oxide nanoparticle reported in the previous investigations. Further, after proper characterization, with characterization, we have performed a, this is proved that the molecules that has been prepared is a biogenic in nature, which has exhibited a few biomolecules that are attached on the surface on the synthesized materials. To investigate their biological properties, we have basically assessed 3 types of property, that antibacterial, antioxidant, as well as the anti-cancer property of synthesized nanoparticles. So as we have discussed earlier also about the copper. So copper nano-generating substantial intermediate due to their competence as an antimicrobial and anti-cancer agent. These properties make them potential candidate for fighting infections and developing a new drug delivery systems. Their presence is already established in various products, including pharmaceutical, cosmetics, and semiconductors. So we have tried that maybe the nanoparticles, because of the biochemical potential of molecules present in cladophora glomata, as per previous study, maybe their presence found in nanoparticle enhanced the activity of copper nanoparticles. So first, we have studied the antibacterial activity by using 2 bacterial strain, 1 gram-positive and 2 gram-negative, Klebsiella and Bacillus, that are food-borne pathogenic strains, by applying agar well diffusion assay, but determining their, and also determine their MIC by measuring their growth rate. So result of well diffusion has showed that the fabricated copper oxide nanoparticles have notably growth inhibitory effect on the both bacterial strains. So whereas large zone of inhibition for synthesis was observed in gram-negative Klebsiella pneumoniae in comparison to gram-positive Bacillus cereus. So after determining their MIC, it is found that it have a higher, you can say, antibacterial activity as comparison to algal extract also. It is demonstrated the present as prepared nanoparticle begin meaningfully overpowering the growth of Klebsiella as well as Bacillus for first tour of the experiment, between the concentration from 10 to 15 microgram per ml, and MIC value for both the organism was found to be 10 microgram per ml. So the obtained result proved that cladophora glomata cell-free extract mediated synthesized copper nanoparticles are suitable bactericidal agent at the concentration of 10 microgram per ml to control the infection of selected foodborne bacterial strains. Further, they control, but we have also tried to work out at what mechanism it's going on for the antibacterial properties. So the 5 bifunctionalized copper first absorb on the cell surface of the bacteria by electrostatic attractions of the organic core of nanoparticles and damage the plasma membrane permeability and gain entry into the cell in order to disable the cellular enzyme functions that cause bacterial death by causing cell wall and membrane disruption that increase the permeability, and oxidative stress that lead to the when it enter to the cells, it causes oxidative stress injury by generation ROS, and that ultimately damage the protein and the DNA and cause the death of the organism. Further, to prove that antioxidant activities has been performed by using DPPH sequencing ability of the synthesized, which is found that DPPH radical up to 50.9 to 80% of selected concentration from 2.5 to 15 milligram per liter. So DPF radical sequencing potential of HNC was found to be 11.25 IC50 values, and total antioxidant capacity was measured. It will be 11 mg per liter. So the overall antioxidant activity result revealed that Cladophora glomata cells free extract also exhibited a good antioxidant activities, might be because occurrence of various phytochemicals on their core, and these electrostatic attraction between functional group of various biomolecules and nanoparticles is probably recognizable for their high free radical sequencing efficacy of biosynthesized copper oxide nanoparticles. So the presence of functional biomolecular entity on the surface of the nanoparticles synergetically increase their bioactivity. Further, after determining their antibacterial and antioxidant activity, which are the promising, and further we have exploited their anti-cancer activity. We know that cancer as an uncontrolled cell. Sir has in first lecture, Sir has explained more on the mechanistic way of or genetics of a cancer cells, but so it is uncontrolled cell growth is among one of the foremost source of mortality throughout the world. And as per the figure provided by WHO, about 25 million individuals around the world have been infected each year, and usually chemotherapy, immunotherapy, radiotherapy, and surgery are used to be cure the cancers. But regrettably, these strategies seem to be more expensive, maybe also affect the unaffected cells and have restricted therapeutic potentials. Therefore, there is an immediate need to develop cancer treatment strategies that are efficient, affordable, non-toxic, negligible side effects, and also acceptable to the peoples. So recent reports suggest that nanoparticles may facilitate the diagnosis and treatment of cancer because their peculiar interaction with having a effective biomolecules on their surface. So we use HepG2, human hepatocellular carcinoma cell line, to determine the in vitro anti-cancerous activity of synthesized nanoparticles. So the synthesized nanoparticle displayed concentration-dependent substantial cytotoxicity on examined cancer cell lines. So the synthesized nanoparticle showed significant impact on cell viability with decrease with increased concentrations. And we found that IC50 value for that is 168.6 microgram per ml. So in comparison to standard drugs that are mostly used to control the lung cancer, that is a felicitations, so that is we have used 2 macromolar. The cytotoxicity of synthesized nanoparticle was comparatively low, but copper nanoparticles synthesized using cell-free extract of Cladophora showed a considerable anti-cancer activity on a HepG cancer cell line as compared to copper nanoparticles synthesized by the other methods. In comparison to negative control, green synthesized copper nanoparticle may have to be ability to diminish 50% of human hepatocellular carcinoma cell line with IC50 value of 168.6 microgram per ml, with certifying its better efficiency. So what is the mechanistic work, that is the mechanism work on the anti-cancerous activity or a control anti-cell by copper. So the anti-cancer active green nanoparticle may depends on a size and shape of the particles. So this is the protective mechanism that we have proposed in our paper, that the small size of nanoparticle allow efficient cellular uptake by cell that facilitate the interaction with intercellular targets. And the copper nanoparticle induce oxidative stress in cancer cell, leading to DNA damage, protein oxidation, and lipid peroxidation, ultimately triggering the apoptosis. And additionally, inside the cell, they also disrupt cellular signaling pathway that is crucial for cancer cell survival and proliferations. So, as conclusions, this is on the basis of our work we have carried on. The present work establishes a low-cost, sustainable approach biofabricating biofunctionalized copper nanoparticles by utilizing cosmopolitan freshwater microalga, Cladophora. The optical, structural, morphological, and thermal characteristics of the biogenic synthesized copper oxide nano have explored using different characterization techniques. Further, the synthesized copper nanoparticle exhibit significant antibacterial, antioxidant, and anti-cancer activity. So the green nanoparticle have demonstrated synergetic activities. So this research could have opened the way in a near future for ecological friendly, economical viable, large scale production of copper nanoparticle by utilizing this green algal species, which you can say we can collect and valorize the biomass by collecting it from nature. And this environmental friendly and economical viable approach for synthesis of provide a versatile platform for development of in therapeutic agents and improve biocompatibility and target efficacy. So this work has been published in one good journal, that is Biosystem Engineering with impact factor 3.6. For here, I would like to acknowledge this work is carried on by my one PhD student, that is Vinay Kumar, and the material characterization work is done by Dr. Sandeep Kaushal. He is one of my chemistry collaborator, and Dr. Ranvijay Kumar has been performed and helped me to perform the anti-cancer activities. So here I will would like to thank the organizer of this conference for giving me an invitation to here. I will try to you can say present one of the you can say work that may be aligned to the main theme of this conference, and maybe I will able to deliver the message to you all, and I will happy to receive any question or queries you have from your sides. Thank you very much. Thank you.
Thank you very much, sir. The session is open for discussion. Students, if you have any questions or query, you can clarify it now. Students, you can ask any doubts, any questions, queries. If you have, you can clarify it now. Thank you so much, sir, for a wonderful presentation on your nanoparticle biomedical application from algal extract. Thank you so much.
Thank you. Thank you.
Thank you, sir.
We now continue with the oral and poster session 2.
Offline poster sessions logs have been pasted. Kindly paste your concerned posters in the respective logs. 10 minutes will be given for your poster. Ready to pin your poster. Students who participated in the offline poster presentation, kindly pin your posters on the allotted logs. 10 minutes will be given.
Students who have participated in the offline poster presentation, kindly paste your posters at the back side of the multipurpose auditorium at the allotted seats. Judges are on the way. 10 minutes will be given for your poster presentation. Kindly pin your posters at the back side of the auditorium. Students, please kindly maintain the decorum. Attendance கொடுத்துட்டீங்க அதனால போறீங்களா?
வீட்டுக்கு இல்ல வீட்டுல பேசுவாதான் வீட்டுல வந்து
Students, I request you to pin the posters according to the given slots.
எனக்கு வந்து லிஸ்ட் தான்
நீங்க அங்க போய் அப்படியே ரேண்டமா ஒட்ட சொல்லுங்க. நீங்களே அதுக்கு அரேஞ்ச் பண்ணி இல்லப்பா அது அந்த லாட்லயே இருக்கட்டும். Dear students, your kind attention please. Those who are participating in poster presentation, please note, I'm going to read out your slot number. I think your class teachers and your guides would have posted the poster list, finalized poster list. So you can go through the Word document. The slot is given in the Word document. But anyhow, I will read out the names and your slot number. So the slots have been allotted there. Below that, you have to pin your poster. Number 1, Hashmita. Hashmita. Number 14. Hashmita, number 14. Trisha, 15. Mohammed Samir, 16. Amelia, 17. Purab, 18. Students, please, may I have your attention? Students, those who are ready with your posters, please post it below the slot number allotted to you. If you do not have your poster, you need not paste. You can leave that blank as it is. Is that clear to everyone? So those who have received your slot number and you are ready with your poster, you can paste it in the allotted space. Poster number starts with lot number 14. Purab, number 18. Kumaraguru, 19. Akshaya B, 20. Angel, 21. Janani, 22. Abdul Gafur, 23. Abina, 24. Ahmad Yusuf, 25. Aravind Raj, 26. Adi Prasanna, 27. Bhardi Kannan, 28. Helen, 29. Bhavani, 30. Celine Infanta, 31. Chandani, 32. Durga, 33. Gopi, 34. Shri Gautam M, 35. Hanok, 36. Kaniha, 37. Harish, 38. Jamuna, 39. Kirtana, 40. R. Kirtana, 41. Kavya K, 42. Kovardhani, 43. Mukundan, 44. Muthusurya, 45. Ajiba, 46. Nishantani, 47. Hemapriya, 48. Pradeep Kumar, 49. Mahalakshmi R, 50. Ranjit, 51. Ritisha, 52. Rohita, 53. Rajalakshmi S, 54. Sandana Krishnan, 55. Shimar Mohammed Yasin, 56. Shrika, 57. Shwati D, 58. Shweta, 59. Divya Shri, 60. Trisha, 61. Tony Crasto, 62. Yogendran, 63. Yuvashri, 64. Hema Malini, 65. Sneha, 66. Mohammed Fazil, 67. Pavitra Haripriya, 68. Jeevashri Kartika, 69. Mahendra Kumar, 70. Murugan, 71. Harish Babu 72, Gunashekar 73, Vaishnavi 74, Carol 75, Jayalakshmi B, Jasmine Preeti, Harsan 76. Participants, I hope all of you are clear with the lot numbers. Exactly 10 minutes will be given to post your posters in the allotted lots. Thank you.
Students, if you have any doubt regarding your slot number, please come to the MC desk. You can clarify.
Ma'am, Suresh, ma'am. Ma'am, online presentation, ma'am. Or share this screen. Ma'am.
Hello.